All right, we're going to go ahead and get started. Thank you for everyone for coming. This is the first time we're on site at the Angeles Clinic after our remarkable, wonderful rebuild, and it's the first time after the pandemic that we're here. So I want to thank everyone for coming. We have a couple hundred people who are Zooming in, and so welcome. I'm Omid Hamid. I'm the Director of Research here. I hope that you'll enjoy the day, a discussion about what's happening in melanoma and the best in immunotherapy and targeted therapies. I wanted to start with introducing Lawrence Piro, who's our Chief Executive Officer, one of the physicians and the dreamer behind the Angeles Clinic. Just for an introduction, and we'll get the day going. Lawrence.
Thank you, Omid, and welcome to all of you. This is a very special 12th symposium, in part because we're back in person after this long COVID adventure, and secondly because, as Omid said, it's the first time we've ever been able to host it in the Angeles Clinic. We founded the Angeles Clinic about 23 years ago, and 8 years ago we joined forces with Cedars Sinai. It's been a great partner to work with, and in this particular conference we of course are partnered with AIM for melanoma, AIM at Melanoma from the very beginning, and a special welcome to Samantha Gill from AIM at Melanoma. We founded the Angeles Clinic on some very simple principles. The idea of having world-class patient care mixed with a world-class patient experience, and a place where research was considered a part of the standard of care, because you can't move the field forward in cancer without asking questions and finding answers. And so this facility that we're in is the manifestation of the world-class patient experience. We've worked very hard to have people who to all of our employees greet patients, whether they know them or not, as if this was their homes. They feel welcome, and we used every bit of surface, light, architecture, art, music, everything we could to create a healing journey. Because when you're feeling sick and you're hoping to be well, you know, that's the healing process, and it takes a lot of connection with your healing places, with your creative places to heal, not just with your medical places. And so I hope that more and more medical facilities will be able to focus on the healing and the artful part, you know, of the journey, because as we hear from patients, it's an extremely important part of going through what is an otherwise, you know, scary experience. So welcome to our home. Thank you for being here. I want to thank all of our speakers. Some of them come from a long distance, and some of them who come regularly to us. They're our friends, and welcome and thank you for you know, putting the time in. And thank you to our speakers from the Angeles Clinic and from Cedars Sinai broader organization. The affiliation with Cedars Sinai has been really great. During these years we've been affiliated, we've soared from, I think, 23rd in the country in terms of the, you know, the various surveys that are done, to being now 9th in the country in cancer at Cedars Sinai, and we're the #1 provider of cancer services in the state of California. So it's been a great adventure, and one that has been punctuated by the simple, continuous commitment to quality, to excellence, and without a mindset of excellence, you can never achieve it, because mediocrity is such a strong force. So every day that we get up and we come to this place, we recommit ourselves to excellence and to try to move the ball up the hill just a little bit more in that day. Congratulations, Dr. Hamid, for 12 years of this conference, and we look forward to, you know, many more in the future. And thanks to all of the people, support people within the clinic who helped organize this and put it together and get it set up. I wish you a very good conference, and I hope that you hear things and learn things that, you know, will transform your lives. And all of our speakers who do amazing research, and those things that we do here, our only hope in life is to transform your life. So welcome and enjoy.
Oh, thank you, Dr. Piro. So it's revitalizing to come to a symposium like this, for me, just to understand that when we began, there was just one drug, and now so many options, and it was so easy to just say we're happy to have something for you. And now it's confusing to say we have so many options that it's become difficult to decipher what's the right way to go and what's the right path to go. And my interest in this type of symposium is to bring the best and brightest to educate not just the patients, but to tell me how to take care of my patients on Monday and our patients throughout the upcoming months. And it's been wonderful to make the relationships like with Dr. Carvajal as the Director of Experimental Therapeutics and the Director of the Melanoma Service at Columbia. So Rick, please come up and tell us about where we've gone with immunotherapy.
Thank you all. I'd like to thank Omid and Dr. Piro for the really kind invitation. And I have to say, this is the first time I've stepped foot in the Angeles Clinic. I've seen pictures, I've heard stories, but this is really a remarkable and magical place, so I'm really happy to be here. So in about 20 minutes, what I want to do is talk a little bit about some of the advances that we've made in the immunotherapy field for melanoma. As Dr. Hamid said, the advances are coming along pretty quickly, and it is really challenging to try to figure out how do we integrate that. What are the lessons we've learned? How do we take these new data? How do we talk to our patients about data? And how do you kind of take that into account when as a team we're making decisions about your care? It's really complicated. Here are my disclosures. And just in the way of background, this is kind of an interesting graph that I like to show that just shows kind of the lifetime risk of developing melanoma in the US over time. You can see that it's really increased pretty dramatically. I mean, more so than almost all cancers. And what I can say, more recently, there's been kind of a flattening of that curve, and so we're not seeing the incidence in kind of the younger patients as much. And so hopefully with prevention, sunscreen, and so forth. Sorry about the feedback. I don't know if I should stand back. Yeah. We'll continue to see a flattening and a decrease in the incidence. But if you look over the past 5 years, there's been, 9,000 or so more cases diagnosed that will be diagnosed this year versus like 5 years ago. But also looking at those numbers on the upper right, thankfully, even though we're diagnosing more cases, the number of people dying from this disease is going down. All right, that's really, really important. And so why is that happening? If you look at the far right, what I have here is the number of drugs that we have FDA approved for different subtypes of melanoma and other skin cancers. I want you to focus there on the right. When we all started doing this disease and treating this disease, we only had a couple of drugs approved. We had chemotherapy, we had IL-2. And so when we saw people in clinic with metastatic disease, we didn't have a ton to offer. And then really starting in 2011, there was just a dramatic explosion of new drugs being developed based on our understanding of the science, biology, immunology of melanoma. And so, these advances, in part, are what have led to that improvement in terms of survival. Okay, and we'll talk about some of those advances. Dr. Sullivan will be talking about some of the advances in targeted therapy. That's been a huge, major component of what we've been able to do. I'll focus on immunotherapy, like what is immunotherapy? Why is this important? And what are the changes that have been made? It really is remarkable. When we talk about immunotherapy, what are we doing, right? It's not like chemotherapy or targeted therapy where we're saying, let's kill the tumor cell itself, right? Let's just attack the cancer. Here, we're giving you drugs, substances, things that are going to change the way your body interacts with the tumor, right? And so as you can imagine, if your immune system was functioning appropriately, right, it would be able to recognize the foreign tumor, right, and just eradicate it. But the cancers are smart. They're going to figure out ways to kind of hide from the immune system. And so what we're trying to do is teach your body to kind of overcome those defenses. And the immune system is really unique, right, in that it's very adaptable. It's got something called memory. And so if we teach your immune system to be able to recognize the melanoma, even as the melanoma changes, your immune system can adapt and create these really long-lasting responses. And that's why we think that immunotherapy, if we're talking about cure, that's our goal. Immunotherapy, maybe is more likely to achieve that than some of our other therapies. So here are some of the critical questions that I think have been answered just over the past year or so. I can't really read that from here, so let me see. Yeah, and so I'm going to spend a little bit of time going through these because, I mean, there have been really key trials that have been completed recently that have answered some of these questions. And so the first one is, we know that adjuvant therapy, that is treatment after surgery, resection of the melanoma, and if there's lymph node involvement, if we give you immunotherapy, we can reduce the risk of the cancer coming back. But there are definitely unknowns there, right? Like what happens in people with earlier stage disease, melanomas where the lymph nodes are not involved. If we treat you with immunotherapy, then will that be helpful? And there's a trial called Keynote 716 that just reported that gives us some data. So I'll share some of that. One of the long-standing questions we've had in the field is, for people who have melanoma that have a mutation in BRAF, so that's about 40–50% of cases, where we have the option of doing targeted therapy that work very well, or immunotherapy, we as a field have not known until recently, is it better to do immunotherapy first? Or should we do targeted therapy first? Or frankly, should we do a combination? And so we have some data to answer that question. The third one is, although we know that treatments like pembrolizumab, nivolumab, ipilimumab, these immunotherapies work really well, we need to be able to figure out how to make the immune system work better. And there are all these new immune targets that we've been looking at. One of those targets that's been of interest for a long time is something called LAG3. And we have new data about targeting LAG3. And then lastly, for our patients for whom we treat with these anti-PD-1 antibodies, that's Keytruda, Opdivo, if the treatment stops working, what do we do then? And we have some data about that. And so just kind of moving on to that first topic, again, we know that immunotherapy, if we give it after surgery for lymph node positive disease, so these are what we call stage 3 melanoma, or patients with metastatic disease after we cut out the sites of metastases, we know that if we give you therapy, that reduces the likelihood of the cancer coming back. Okay, so that's well known. But what are the unknowns? One, that immunotherapy, that adjuvant therapy will help some people, it won't help everyone, right? We only want to give it to the people whom it's going to help. And I don't know, we don't know who those people are, one. And two, what do we do about earlier stages of disease? And so Keynote 716 was a trial where patients with stage 2 disease, node-negative disease, right, that were 2 millimeters or deeper, ulcerated, were randomized to either pembrolizumab or placebo, okay? And I'm just going to talk about that part 1 that's kind of there in red. An important part of this trial is part 2. That is for the patients who are treated with the pembrolizumab or placebo, if the cancer comes back and they're treated with Keytruda later, what happens? Okay, and I'll come back to that in a little bit. The primary endpoint of this trial was recurrence-free survival, right? So we don't, which drug is better at preventing the cancer from coming back? And as you can see on this curve, the immunotherapy is the top line, that better line. And if you look at that 12-month improvement, there's a very real improvement in the 1-year recurrence-free survival. And I think this is pretty compelling to tell us that it in fact does work. If I give you immunotherapy, no negative disease, I can reduce the risk of the cancer coming back. But that comes at a cost, right? And that cost is the side effects. I think, I suspect many of you are on immunotherapy and maybe have experienced some of these side effects. Some of these side effects are acute things like diarrhea, inflammation of the lungs that, it's bad when you have it, we can usually fix it. Some of these side effects are kind of maybe not super severe, but not insignificant. And that's kind of fatigue and joint aches and just not feeling well. And then another class of side effects are these, what we call endocrinopathies. And these are the side effects where some of the hormone glands of the body are shut down. And the issue with these is these side effects, if they happen, are permanent, right? If I shut down your thyroid function, you have to be on thyroid replacement therapy lifelong. And so there's a certain kind of extra significance, I think, to this class of side effects. But on this trial, nearly 20% of patients treated with the immunotherapy developed these permanent side effects. And so, what do we know? We know now that anti-PD-1 therapy in stage 3 disease, node-positive disease, metastatic disease after resection works. We know now that in high-risk node-negative disease, so now that's what we call our stage 2B and 2C patients, it also works. But what we don't know is, are those toxicities worth that potential benefit? And I put this unknown because right now I don't know if it's better to treat with adjuvant therapy now, which I know will reduce the risk of it coming back, or if we say, let's watch you carefully, and if God forbid it comes back, let's treat you then and potentially spare you the risk of those side effects, the outcomes may be the same because we're now able to cure, not everyone, but some people even with metastatic disease. Okay, and so, part 2 of that keynote trial will help us answer that question. I want to shift to kind of metastatic disease, and this is kind of just a timeline of what treatments we had available and what we thought about treating people in the setting of metastatic disease, frontline therapies. And in 2010, we had chemo, we had IL-2. In 2011, that's when we started to get the approvals of these targeted therapies and these immunotherapies. And that's when we started really breaking down our management of patients into their kind of distinct molecular subtypes, right? And there are a lot of molecular subtypes, but one of the bigger ones that we think about are, do patients have a melanoma that has a BRAF mutation, yes or no? And for folks without a BRAF mutation in 2011, we were treating with ipilimumab, that first immune checkpoint inhibitor. If you had a BRAF mutation, then you had the option of targeted therapy as well. But you can see in 2013, there were further advances. We were using not just one targeted therapy, but combinations for BRAF mutant disease. In 2014, we got the anti-PD-1 therapies approved. In 2015, we started looking at combination immunotherapy. And in 2015, really what we had for a BRAF wild-type disease was either ipilimumab plus nivolumab, combination immunotherapy, or single-agent anti-PD-1. And for our BRAF mutant folks, we could do targeted therapy with a BRAF-MEK combination. And again, if you go back there for the BRAF mutant patients, until recently, it wasn't clear, do we do immunotherapy or do we do targeted therapy? To help answer that question, there was a really incredible trial that was called the DREAMseq trial. Really challenging trial to do, but basically what this did was took patients who had a melanoma with a BRAF mutation that was metastatic and randomized them to either get immunotherapy or targeted therapy to try to help answer that question. And if the cancer grew, they would get the other, right? And so this is kind of the question, does sequence matter? This is the data looking at response rate, that is, what's the likelihood of shrinking the tumor? Let's look at the top part, the top 2 bars. Arm A, if you got immunotherapy, there was a really high response rate. We're talking 40, 50% response rate. And Arm B, if you got targeted therapy, same thing, a really high response rate. What was interesting, though, is if you got immunotherapy first, Arm A, and then if the cancer grew, you got targeted therapy, that's Arm C there, the response rate to target was about the same, right? It didn't make the responses any better. It didn't seem to make it any worse. If you got targeted therapy first, which is that Arm B, and then at the time of progression switch to immunotherapy, that's Arm D, it seemed like the responses were a little bit less robust. Right? So from a tumor shrinkage standpoint, it seems that immunotherapy first may be better. And then if you look at the other important endpoints, like how long can we keep the disease under control? Clearly it's better if you get checkpoint blockade first. And so really, our practice now for almost all patients is to do immunotherapy first, and then save targeted therapy for next line. That's not going to be for all cases, so that's going to be true for the right answer for many, many people. Now, just a few months ago, we got another drug approved, which is a combination of relatlimab and nivolumab. And relatlimab is a drug that blocks something called LAG-3. It's another one of these immune checkpoints. And that LAG-3 protein increases on the tumor, on the immune cells in cancers, as the the immune cells kind of tire out. And so we call it an exhaustion marker. If we block that LAG-3 thing, we can rejuvenate the immune system and it can function better. And so in the Relativity Trial, patients were randomized to either the combination of what we call Rela plus PD-1 versus PD-1 alone. And the primary endpoint here was disease control. If you look at the tumor shrinkage rates, and it's really the top line there, about 43% of patients got major shrinkage if they got the combination versus about 32% for the single agent, suggesting the combination is better. And then if you look at a disease control, which is this progression-free survival endpoint, it's also better if you get the combination, okay? One of the outstanding questions in the field, though, is, you know, Rela-Nivo seems to be better than Nivo alone in terms of efficacy, but what does that mean when we compare it to Ipi-Nivo, the other combination we tend to use? You know, and if you do these, what we call these cross-trial comparisons, the efficacy still seems to be better for Ipi-Nivo. We've got longer follow-up for Ipi-Nivo, and you know, if there are questions from the group, I think all of us will answer it a little bit differently. You know, but the data in terms of what is the treatment that's likely, that gives us the highest likelihood of curing disease, we have the most data for Ipi-Nivo. And so, you know, now for immunotherapy, we have three options. We have either single agent PD-1, which can work and can work really well in some people. Clearly the best tolerated, right? And you know, if I knew who single agent PD-1 was going to cure, that's definitely what I would do. Second option is Ipi-Nivo, the combination we've used for a long time. A lot of data. We know we can cure a good fraction of people with that combination, but comes at the cost of side effects. And then Rela-Nivo is kind of like an in-between. It works better than single agent PD-1, maybe not as well as Ipi-Nivo, but with fewer side effects. And so now we have to figure out, you know, how do we take that data, integrate it into our patient care? And some of this is going to be biomarker driven, and so there's still more work that we need to do to kind of figure that out. Now, the last bit I want to talk about is, let's say people were treated with PD-1, Keytruda, Opdivo, and the cancer grew. What do we do? Outside of a trial, what do we do? And this important trial by SWOG, the melanoma group which Dr. Patel runs now, helped to answer that question. And so here, patients who had single agent PD-1, who really didn't have a response to that treatment, were randomized to either the other immunotherapy, ipilimumab, or the combination of ipilimumab and nivolumab, right? To see, do you need the combination or can you just do that one immunotherapy? And in terms of tumor shrinkage, you can see that, and this is a waterfall plot, right? Each bar is a patient. If the tumor shrinks, the bars go down. You want everything to go down. More bars go down for Ipi-Nivo on the left than on the right. So in terms of tumor shrinkage, it's better for the combination. Primary endpoint of this trial was, again, progression-free survival, duration of disease control, also better for the combination. And so this basically tells us that if someone is treated with Keytruda alone, Opdivo alone, and the cancer grows, if we're going to do standard of care treatment, it's going to be Ipi-Nivo. So just to kind of summarize, one, does adjuvant immunotherapy work in lymph node negative disease? It does, and we can do it. It's not clear if it's better to treat earlier, though, or wait and see if, God forbid, the cancer recurs, treat later. Two, do we do IO first or targeted for BRAF mutant melanoma? For most cases, it's going to be IO first. Three, does the combination of LAG plus PD-1 improve outcomes? And it does. I do think that we have more data. We do have more data for Ipi-Nivo, but we have to, I think, as a field, figure out how we're going to use this new tool that we have appropriately. And lastly, in melanoma that grows to single agent PD-1, is it better to do Ipi-Nivo or just Nivo alone? And here the answer is combination. And so I'll just close with this slide. You know, there's a lot of work that we're doing right? To try to understand, why does our current immunotherapy not work? How does the immune system, how does the tumor evade the immune system? There are all these mechanisms, right, that have been identified, and this is just some of them. And there are trials now kind of trying to address all of these issues. And so, I just took the family to Disney. We did the Star Wars thing. The force is really strong with the field. We've got an incredible collaboration of investigators, clinicians, scientists, patient advocacy groups like AIM and Melanoma. The patients and caregivers are super passionate. And I think we're going to continue to make progress. It's never fast enough, but I think we're going to get there. We will get there. All right, so I'll leave it at that. Thank you.
Okay, so back to some housekeeping. Those of you who have questions, please hold them. We'll have a question and answer session at the end. There is coffee and pastries out there. Please feel free to step out as you want. We'll have a little bit of a break soon. Then we'll move on. But afterwards, again, these talks will be available to you through the AIM link, and if you connect with us at Angelus. What I forgot to do is to shout out everyone who is joining us. This Zoom and Microsoft Teams and everything has become such a part of our lexicon that we forget how amazing it is to bring people from all over the world. And as you can see, I'm not the most tech-savvy person, but I wanted to shout out the people from all around the United States and multiple different countries who are coming in here. I remember when we did our first one of these in 2011, and people flew in, so we had to do it next to LAX. That was the thing. We needed to find a place next to LAX because people said, oh, we're going to get on a plane and come. How crazy is that now? And we had about 100 people, and about 10-20% of them had come from outside of the greater Southern California area. Now, thankfully, our issue is, well, should we even do it on site? Because it's such a global community. And that's phenomenal. But it's also great to bring friends in so that we can share time together and we can collaborate. And that's really what medicine is about. It's the proximity to your colleagues. It's about the proximity to the patients and the ability to discuss honestly what's going on and be happy and educate each other. So that's why I would always insist on, even if there were 2 people that showed up here, having something here. And why I'm proud and happy to have a very good friend. We're going on our 9-year anniversary of being friends and colleagues. This is Sapna Patel, who's just a meteoric rise. She's the chair of the SWOG Melanoma Committee. That data you just saw there comes from SWOG, comes from her leadership, and also works as the director of the uveal melanoma program in a very, very small place you might have heard about, MD Anderson Cancer Center. I hope that she'll enjoy her time here. She'll take something of what she's learned about Angelus and teach it to the MD Anderson people. But truly a pleasure to have you here.
Thank you, Omi. That was such a lovely introduction. I'm going to swing myself around so I can see the slides a little better. Oh, no, maybe I won't do that. Actually, MD Anderson just celebrated its 80th anniversary, and it looks and feels 80 in some parts. So we actually really could learn about what it looks like to modernize and improve the patient experience. Okay, I'm going to put a timer on me so I don't just ramble and ramble. So I'm going to talk to you guys about neoadjuvant therapy, which is medicine that you would take before surgery, and why this is a path that we're exploring in melanoma. So in full disclosure, I am the National Study Chair of SWOG S1801, which is currently the largest completed neoadjuvant trial in melanoma. So that's a disclosure that's relevant for this conversation. So the rationale for neoadjuvant systemic therapy, sometimes it's abbreviated as NAST, is that in the top cartoon, if you were to remove a tumor, there might be some microscopic tumor left behind. Let me see if I can point to it here. There's some little tiny tumors left behind. That's microscopic cancer. The surgeon can't see it. They don't know that there was anything left behind. And then we infuse medicine, those Y-shaped drugs. That's essentially what immunotherapy is, or these antibodies. And what can happen is that immunotherapy is trying to get your body to create T cells against those microscopic cancers. And it can do a pretty good job. These T cells here. As Dr. Carvajal noted, in the adjuvant or resected setting, it works for some people. Your body does generate T cells. But the idea behind leaving tumor in place and doing the medicine first is that you will get a larger expansion, a larger number, and a larger diversity of T cells if you leave the tumor in place, because now it's just not the microscopic, it's the whole cancer, the whole tumor, and your immune system will build up against it. You're still going to remove that cancer, but what's left behind is a much more rich and diverse T cell population. So the idea is if somebody has resectable melanoma, you can take it out. Taking it out is still on the table, but perhaps doing immunotherapy first will give you a longer, richer response on the back end. And this was actually proven in a trial called the Opposin trial out of the Netherlands Cancer Institute, where they had a resected melanoma arm and then a neoadjuvant immunotherapy arm, and then patients went to surgery. And what you see in the bar graphs here is that in the adjuvant arm in red, compared to the blue bars, at first glance, the blue bars are taller. So that's number of T cell clones. You get more T cell clones if you do medicine first. And then in the light red and the light blue, and the first one's a little distracting because it's so tall, but if you look at the majority of the others, the light red bars portions are shorter than the light blue. That's the number of new clones that were present after surgery. So the surgery happens and how many new T cells did you develop? And so that's very interesting that leaving tumor in place gives you numerically more T cells on the back end and perhaps more clones, so a richer diversity. So they actually proved it. And what they proved as far as survival is that if you took neoadjuvant therapy, which is the curve in kind of gray there, that you had a better recurrence-free survival, is the term we like to use in the adjuvant setting. You have a better recurrence-free survival compared to doing surgery and then possibly taking medicine after. This study was very, very small, though, just a handful, few dozen patients. So there wasn't necessarily a difference in the long-term survival, which may be a good thing as a patient, right? It means whichever path is chosen, surgery first or medicine and then surgery, it's okay because in the long run we can catch up your survival, whichever treatment you took. But we do like the diversity of T cells that come out with neoadjuvant therapy. So the Opposin trial was very what we call hypothesis generating. It was an early proof of principle, but it was a very small study. So you always need to validate these things in bigger studies. But internationally, what was decided is perhaps we need to have a consortium of doctors. So what they're doing in Australia, in Europe, and in the United States is harmonized. And we're not doing things 3 different ways and then trying to draw conclusions. So the International Neoadjuvant Melanoma Consortium was formed. And I feel like they might have stolen this picture from Dr. Sullivan, or maybe it's the inverse. So this is a graph of along the way when somebody's getting neoadjuvant therapy, what do you do with the tumor? When do you collect blood? And perhaps when do you also collect what we call microbiome, mouth swabs, stool samples, et cetera, and you collect them periodically along the way to learn things. It was important to harmonize this up front, otherwise you would have Europeans collecting at different time points and Australians and Americans, and we have seen global differences in treatments, and you often wonder if it's just due to a slight difference in practice. So the International Neoadjuvant Melanoma Consortium was formed and said, roughly, we're going to give about this much neoadjuvant therapy, somewhere around 2 months worth of neoadjuvant therapy, and then try to have surgery between the 2 and 3 month mark and then follow on. So through those guidelines, the International Neoadjuvant Melanoma Consortium went and pooled a bunch of data out of the United States. The first trial was run at MD Anderson using targeted therapy. I can't see that one. That was out of Australia, the next one out of Netherlands, another MD Anderson using immunotherapy, the University of Pennsylvania study using 1 dose of immunotherapy, just a single cycle of pembrolizumab, and then a study using just immunotherapy with no adjuvant therapy. And you can see already there's a variety. The yellow bar is how much neoadjuvant therapy. Everybody got a different amount of weeks of neoadjuvant therapy, but roughly in that 2 to 3 month mark. But everybody got a different variety of adjuvant therapy, some up to a full year, some half a year, some a few. So all of this needed to be harmonized. But importantly, when you pool this data, and pooling will have some flaws because the patients who enrolled are different, characteristics are different. So we take all of this data with a big grain of salt, but when you pool the data, you find that the immunotherapy patients end up having a stronger survival based across many different trials than perhaps the targeted therapy. So if there's a BRAF positive melanoma, as Dr. Carvajal said, you have options. You have options between BRAF pills or immunotherapy infusions. But even in this setting, it looks like, apologies, everybody's getting that alert. Even in this setting, it looks like immunotherapy may run the day here. And so higher survival curves with the immunotherapy and targeted therapy. And what we see is, again, with neoadjuvant therapy, you can get single agent immunotherapy, or you can take that immunotherapy in combination with more side effects, running the risk of missing your surgery date because you're dealing with side effects. So with all of that consideration, it does still look like more immunotherapy upfront might be better, but very small data sets here that we have to validate, essentially. This curve overlaps what if you just went to surgery first? If you went to surgery and took your adjuvant PD-1, single agent, it's interesting how that overlaps with the neoadjuvant PD-1 curve. So that's a very interesting observation, and it proves the point again that we have not compared adjuvant head-to-head with neoadjuvant. We have the very small Opposon study that, put this seed in our head, was proof of principle, but we've not actually proven that neoadjuvant is better. And so there are 2, what I would call milestone or landmark trials that will help answer this question. And as I disclosed, S1801 is really one of them. This is a study of up to 500 patients, but actually we stopped at about 350, 360, because we had enough events occurring that randomized patients 1-to-1 to either traditional surgery and 18 doses or a year of pembrolizumab every 3 weeks, or 3 of those pembrolizumab doses, then surgery sometime between months 2 and 3, and then 15 doses of pembrolizumab after. So there's balance in both arms between the number of pembrolizumab doses. There should be balance between the surgery, both arms getting surgery. It's just a question of, does surgery up front actually do anything to improve your outcomes? And so the outcome of this study is what we call event-free survival. How long does a patient live on each arm until something happens? And that something is defined as their disease recurring or progressing. So if you're on the neoadjuvant arm and your cancer actually gets bigger on treatment, because we know immunotherapy does not shrink 100% of tumors, did your tumor grow in a way that it's no longer operable, that you actually lost your operable window? That would be important to know how many times that happens when you give pembrolizumab before surgery. What about patients who do surgery, but because they go to surgery first and it ends up being a big, bulky surgery with lymph nodes and so forth, they have problems with wound healing and they need wound vacuums or other things. They never actually get started on the adjuvant therapy. That would be considered an event, and it might give us thinking to use neoadjuvant therapy if the tumors shrink and the surgeries can be minimized so that there isn't this postoperative waiting period for healing. Anytime after recurrence, say both arms go to surgery successfully, finish their year, any melanoma recurrence after that is an event. And then because this is the S in event-free survival and progression-free survival and overall survival accounts for being alive or not, then death is another endpoint. And it's death from any cause, which I have issues with. It should be melanoma specific. But this study actually closed to enrollment about a week ago, and we're very happy to announce that. That means that we didn't need to take 500 patients and randomize them because we're getting the answer sooner, but I have no information on what that answer is going to be. We haven't combed through the data. We hope to have information later this year. And one of the things we learned in 1801 is you actually have to gross the specimen properly. When you do a neoadjuvant pathologic response, it's a little bit different than just opening a tissue and saying, is there cancer or not? That's a lot of times the only question we're asking on a biopsy or a surgical specimen. Is there cancer and kind of how much? But on a neoadjuvant, you actually need to get the entire tumor bed and ask the question, how much is dead tumor and how much is alive? So it's not simply is there melanoma and measure how much, it's how much on a denominator of how much was there. And that requires grossing tissue very specifically. If you have, these are lymph nodes, they're black because they have melanoma involved. If you have a lymph node less than 5 centimeters, it requires grossing a tumor in a very specific way versus if you have it greater than 5 centimeters. Usually less than 5 centimeters, we gross every few millimeters. And then if it's greater than 5 centimeters, because that would make hundreds of slides for us, we end up grossing at 1 centimeter hashes. So we learned this as we were developing 1801, that we would get tumor blocks and it would give us the tumor size, and we would realize, well, it was a less than 5 centimeter, but we actually didn't get enough slides here. It should have been grossed more cleanly so we can actually say how the tumor responded. Similarly, here's one that was small. It doesn't look like we got enough tissue. So it was very iterative, this process, where we went back and taught sites, here's some information to post in your surgical suite and in your pathology suite. Make sure the tissue is grossed in a specific way so the pathologist can do a neoadjuvant read. That's very important. It's not just a regular pathology read. So we just have some guidelines here that I think were helpful for sites. The other landmark trial is the Nadina Trial. This trial is being run out of the Netherlands, is involving, I think, most of the Netherlands, maybe also some parts of Europe. And what this study is doing very similarly is saying you'll get some neoadjuvant immunotherapy, a therapeutic lymph node dissection, because these are mostly for patients with lymph node involvement, or you'll get a therapeutic lymph node dissection and then your adjuvant immunotherapy. Interestingly, they have a few extra variables in this trial. So after the therapeutic lymph node dissection, if the tissue is grossed properly and it looks like the majority of the tumor is either dead or gone, or all of it is gone, major pathologic response, pathologic complete response, or near pathologic complete response, then they could actually not do any adjuvant therapy. So they could stop right there and be done. If they have no major pathologic response, so more than 50% of the tumor is still alive and kicking, and they have a BRAF mutation, it can be BRAF adjuvant therapy or immunotherapy, so that's a variable in there. And then if they don't have a BRAF expression, then they would just do immunotherapy. Very interesting design. This study will probably be finished in 2023, so we'll hear results maybe next year or the following. And one of the things the Netherlands did that was very interesting across many of their opposite trials was they looked at something called an interferon gamma signature. So when a melanoma diagnosis is made, you guys know there's a biopsy that's taken. In that biopsy, you can run a series of genes for a cytokine or a signaler in the body called interferon. And based on that interferon signal and potentially tumor mutational burden, but in data that I can't share right now, it looks like interferon gamma signature may be sufficient. It looks like there are people, the curves essentially separate, and you can say there will be responders to the neoadjuvant therapy, and there'll be less favorable responders. They're still responding, maybe just less favorable. So on the basis of that data, the Netherlands group, that's a very prolific neoadjuvant group, they've developed the Donomy study, and the Donomy study says we'll take that pre-treatment biopsy, and in 2 days, We're going to tell you if you have an interferon high or low signature. I feel like we could never work that fast here in the United States. In 2 days, the tissue has like barely moved out of the warehouse. But in 2 days, they're going to know if you're interferon high or low. And based on that, you're going to get randomized. So if you have an interferon high signature, you're going to get single agent. You're going to get single agent PD-1 because that's what some of their preliminary data suggests. You might get it in combination with this dometinostat. Dometinostat is an HDAC inhibitor. And if you're interferon low, you're going to get dometinostat with that Nivo, or you're going to get what you should be getting is combination Ipi, Nivo, dometinostat. And then everybody goes on to have a therapeutic lymph node dissection, et cetera, et cetera. It doesn't look like they're doing adjuvant therapy here. So that's very interesting. They're using that pre-treatment interferon gamma signature. And then across multiple trials, what they found is that if you have an interferon gamma high signature, it looks like you might do well with Nivo and Nivo-Ipi. But if you're going to do well with Nivo, you may spare yourself the toxicity of the Nivo-Ipi and just take single agent. In the interferon low, interestingly, it looks like double is giving you some responses, but they're still suboptimal. So in low patients, we're brainstorming, should that be triple therapy or something else? And then one of the last slides I have is about changing surgery in response to your neoadjuvant therapy. So what they're doing in the Prado study in the Netherlands is giving patients neoadjuvant combination for 2 cycles. Then they'll take the index lymph node and under the microscope determine, did you have a near response, a partial response, or no response? If you have a near response, you don't have to go through with that therapeutic lymph node dissection, and you'll just be monitored. If you had a partial or no response, then they will do a therapeutic lymph node dissection and followed by adjuvant therapy depending. So that's an interesting strategy in neoadjuvant to basically personalize the response. So neoadjuvant therapy is really intriguing. In small studies across many countries, there's been a signal, but we really need a head-to-head trial against adjuvant therapy. We need to demonstrate that neoadjuvant therapy is actually better than what we've been doing. And S1801 and Nadina will be the 2 kind of milestones. Once those finish and report, we'll have a couple of studies now to discuss. And there are opportunities to improve the extent of surgery with the Prado study, perhaps the choice of neoadjuvant therapy with the Nadina based on an interferon. The choice of adjuvant therapy based on Nadina, which was BRAF or targeted therapy, and the choice of neoadjuvant based on the Donomy for interferon signature. Pathologic evaluation of the surgical specimen we think is a really important step, and it's not the same as pathologic evaluation just after surgery alone, so this requires special attention. And 1801 and Nadina are going to be co-analyzed together to look for that interferon gamma signature so we can actually see is it helpful in 2 different studies using similar models. And that's it. Thank you, guys.
Okay, thank you for that lovely talk. A couple of things. We will move this podium after our break, number 1. Number 2, we got patients who are sitting here going like, why did I get my surgery? And they're going to line up and want to. So the neoadjuvant paradigm is one that's coming. You do really need to have a lot of tumor so that we know that you're a stage 3. Some patients we can't tell if they have lymph nodes involved, and therefore they go directly to surgery, and that's appropriate. I did want to say that the Europeans are running Nadina, but also the Americans are running it. MD Anderson will be doing Nadina shortly, as will the Angeles Clinic, a plug for the Angeles Clinic, as we move forward, and a couple of other sites in the United States. So it's a good transition when you start talking about neoadjuvant therapy, because this is now transitioning into the surgical oncologist portion of this meeting. So neoadjuvant therapy is not taking the surgery away from the surgeon. It's just helping to understand how to best take care of patients. One of the most amazing things that we're going to get from neoadjuvant therapy is data from tissues. How were the tissues before? What did our treatment do for them later? Who responded, who didn't? And then even before someone sees a surgeon or sees an oncologist, the pathologist can help us to say what kind of pathway they really need. So that transitions well into my discussion about Dr. Mark Ferries. And Dr. Ferries and I have been together for almost a decade and a half. And truly it's a melding of surgical and medical oncology, because a lot of this, you feel like the medical oncologists are pulling the surgeons into it, but it's truly the opposite. And Mark has been wonderful in educating the medical oncologists about why these things are so important and forcing us to bring these therapies on and build these therapies into our programs, specifically here. It's harder to build these programs. It has many moving parts, but Mark's a taskmaster, and I'm very afraid of letting him down. But I hope that everyone in their career and in their jobs finds a partner like Dr. Ferries. When I thought about who would talk about TIL therapy, I realized that Dr. Ferries has run a TIL program even before they became, in vogue. He was a icon in T-cell therapy in this area in LA, because even before others were doing it, he was doing it almost with his sheer energy. So I'm going to ask that energy to get up here and talk to us about TIL therapy.
All right, thanks very much. Dr. Patel mentioned that it's MD Anderson's 80th anniversary, which is an incredible coincidence because Dr. Hamid and I started working together 80 years ago exactly today as well. So it's tremendous. But thank you for being here. Thanks for everybody joining online. So my job is to tell you about TIL therapy, which is coming on strong. And the first task part of that is, what is TIL therapy? TIL stands for tumor infiltrating lymphocytes. It may or may not be evident, but so many of the things you're hearing about today center in one way or another on lymphocytes, white blood cells. So that's a lymphocyte, and it is in many ways the key to the effectiveness not only of TIL therapy, but a lot of the other therapies that you've heard of. It's not the only part of the immune system, and there are proponents for a lot of other, for B cells and macrophages and other things, but they have taken the lead in the last few decades. So what do we talk about? Why do we think the immune system can fight cancer? And how do the lymphocytes do this? And then making this work in terms of overcoming resistance that tumors can have to immune attack, and how can we essentially manufacture an immune response for patients who need it, and then what's on the horizon. So we know that the immune system can recognize and attack melanoma cells and other tumor types. And we know that, let's see, my letters are not there, but that's all right. So this is just a microscopic image of a melanoma. And the big pink cells on the top are melanoma cells, and the small round blue cells at the bottom are lymphocytes, these white blood cells. And this is before any treatment. This is just a biopsy, basically, that a patient had of a melanoma on the skin. And what it tells us is that somehow the body on its own has recognized these cells as abnormal. And they have mounted this response, and these cells, which would not normally be there, the lymphocytes, are working their way in the hopes of attacking those abnormal cells and getting rid of them. And unfortunately, they're not totally successful in doing that all the time, but this is a real indication that this is a natural phenomenon that we may be able to take advantage of. And so this goes way back in terms of using this immune response to a guy named William Coley, who was a surgeon in New York at the end of the 19th century, and he noted that a few patients that had Tumors, if those tumors got infected, in some instances the tumors then went away. And so what he thought was, well, why don't we introduce some products of this infection to the tumors and see if they'll go away with that? And indeed, he had many examples where that actually happened. They injected bacterial toxins into tumors, and they dosed to how much they were supposed to give by when the patient had a fever of 102. That was enough. But it was difficult to reproduce, and the science wasn't there, and so it sort of fell by the wayside as chemotherapy and radiation came along. We have used similar techniques in using immune treatments even before this raft of new great medications that came along. And so we used to inject tumors like this with something called BCG, which is a TB vaccine. And in this particular patient, we also applied a topical cream called Aldera to it together with the BCG injections. And over a series of a number of weeks, the tumor all went away. And he had no side effects, and he was in his 90s at the time and went on to die of something else a number of years later. So we've known that this can work. One of the things that we love about immune therapies is how durable the response can be. This is a very old study of one of the original immune therapies called interleukin-2. And although the response rates to interleukin-2 are not very high, the people that get a response can get a response that lasts forever. And so the important aspect of this survival curve, and this is a curve where as people die, the curve drops down, you see that after a bit of time, the curves are flat. And they never drop down again, meaning once you've achieved this response, it can last with you forever because you carry your immune system with you forever. And so the situation has changed from the old IL-2 days. This is another version of that slide that Dr. Carvajal had. And what you can see is we had almost nothing for a very long time, and then there's been this explosion of new drugs, effective drugs, much more than what we had before. And all the things that are in blue and the things that are in white there are immune-based treatments. And so it's a very different world to what we had before in terms of the number of options. But one thing that has been consistent with these immune therapies is this flat curve. So ipilimumab here, you can see that after a couple of years, the curve is flat, and over many years then it never drops down again. This is a summary of some of those neoadjuvant studies Dr. Patel was talking about. Flat curves then again, at least in terms of early follow-up. So that's why we love, one of the reasons why we love immune therapies. So how does all this work? I have no idea, right? I'm just a surgeon, so the smart people you'll hear from later and they may explain it to you, but this is my simplistic cartoon understanding of some of these things. So if you have a melanoma, there are antigens, there are targets for the immune system that are in the melanoma, and either released on its own or when the melanoma dies, these antigens are released or exposed, and this is about potential targets for the immune system to recognize and attack. And then there are what are called antigen-presenting cells that take up those antigens. Those cells can then mature into things that are able to present those targets to the lymphocytes. So the lymphocytes then can see the targets, get energized to go off, and kill the melanoma. And the cycle hopefully then continues. And the cycle can be influenced at a number of spots with treatments that we can impose and with other components of the immune system. So there can be helper lymphocytes that help this response go. There can be regulatory cells that block it. And the hope is that these little blue lymphocytes will go kill the big tumor cells and then move on and kill more tumor cells. And it's often talked about as sort of like driving a car, the components of this immune system. And so what are those components? Well, you can think of this energizing of the antigen-presenting cells as sort of the ignition switch, right? When we see danger, the immune system is turned on, and then we produce these immune responses. You can think about these things on the lymphocytes, T-cell receptors, as kind of a steering wheel. It tells the lymphocyte what it's supposed to attack. It identifies these targets. And you can think about this interaction between the immune cells and the melanoma cells as something that might have things that step on the gas or that step on the brakes. And so you can have helper cells, regulatory cells. You can have these blockers, CTLA-4 and PD-1, that block the ability of these cells, these responses to be generated, and other things that can advance it or put gas on it. And so these have then enabled many people to develop some of these therapies, which you know about. So, for example, one of the brakes is CTLA-4. And how does that work? So if you have an antigen-presenting cell and you have a lymphocyte, the normal way that these two things interact is the lymphocyte comes up and the antigen-presenting cell shows it a target in a very specific sort of context. And then there are co-signals, in this case CD28, that is on the lymphocyte. And when these two things come together with the target, it turns that lymphocyte on. And then the lymphocyte goes off to kill the stuff it's supposed to kill. Eventually, the lymphocyte may come back into contact with that antigen-presenting cell. And now at this point, they've lost that CD28 that turned them on, and they've gained something called CTLA-4 that turns them off. And so that lymphocyte is turned off. This is kind of like a parking brake. So ipilimumab, what it does is it keeps this from happening and releases this brake. The other version of this is sort of the foot brake. And so this is the PD-1 based drugs. And here it's an interaction between a lymphocyte and a tumor cell. And the same target receptor interaction that would tell the lymphocyte this is something that has a target we're interested in. But there can be this interaction between PD-1 and PD-L1 on the two cells that then says, yeah, don't attack this cell, leave it alone, and it shuts that lymphocyte off. So all these PD-1 drugs that you know about work right here and keep that from happening. And there are not just those 2 points that we have interest in affecting how these the treatments work. In fact, there are many points along this relatively complex cycle at which we can potentially intervene and help move things in the right direction. So we can kill more cells with radiation, with something called TVEC that you may know about, an injected treatment like the BCG. We can help turn on the ignition switch with things that activate those antigen-presenting cells, vaccines, those sorts of things. You can pour gas on it with interleukin-2, and there are lots of other places along the way that we can potentially have drugs that would help with this response. And so the question for simple-minded me is, is this really like driving a car, since it's a little bit more complex? And the answer is no. It's probably more like driving a space shuttle. This is where all this excitement comes from that we have, and where all these trials that you've heard about with different agents and different drugs come along. So I'm not really supposed to be talking about any of that. I'm supposed to be talking about tumor-infiltrating lymphocytes. And so back to T cells, it's not this complex thing where we have to be flying the ship as it's moving through this thing. Basically, what we do is we create these lymphocytes in the lab and then release them to go off and do their thing without us necessarily having to intervene in the same way, so more like a cruise missile. So how does this work? We know already that lymphocytes that are in the body can recognize and infiltrate into tumors naturally on their own, part of the body's normal immune defense. And what we can do surgically is take out a tumor and bring it into the lab, where then we can put it into conditions where we can see that it has lymphocytes in it. You can identify them with special stains that really highlight them. But we can put those pieces of tumor into culture conditions that then allow the lymphocytes to have an advantage. And they grow up and proliferate. Eventually, they kill the tumor that's in the wells in the culture in the lab. And then we can take those lymphocytes that have grown out and test them against tumor targets and see which of these subcultures of cells actually kill those tumor targets or react to them in the lab. And then the ones that react, we can put through what's called a rapid expansion protocol. We've used these special gas permeabilized flasks, but it really allows you to magnify the number of these cells thousands of times to the point where you have billions of these anti-melanoma lymphocytes that then you can give back. So you take those and you give them back through an IV that go into the patient, and then they make their way around the body, hopefully to find any melanoma and kill it. There are special conditions that have to apply for when these treatments are given. So one is you have to get rid of most of the lymphocytes that are in that patient before you give these cells back, essentially to create space for those cells then to occupy when they go in. So there's chemotherapy that's given to do that before the cells are given. And then this interleukin-2, which is a white blood cell growth factor, is given to help support these lymphocytes once they're infused and help them grow and proliferate. And those are actually the toxic parts of this treatment and what make it so challenging. The lymphodepletion is a little bit toxic, and the interleukin-2 can be quite toxic. So not every patient is eligible or is a good candidate to tolerate this, at least as it's given in the standard way. But does it work? We know it works. Dr. Rosenberg at the National Cancer Institute has been the pioneer, has been the patron saint of this for many, many years. And they've shown repeatedly that it can work. So the tumors disappear, they disappear, they disappear in patients that respond to this therapy. But it has always been thought, or it was thought for many years, that this couldn't be done outside of the NCI, very specialized center. So this is Google Earth view of the NCI. It's a small city, right? There's another place that you may have heard of, again, called MD Anderson, also a small city. So this is the kind of place where this stuff was done because they had these considerable resources and specialists to be able to do that. But it actually doesn't take a city, it takes a village to do this. And Dr. Hamid and I worked together on a protocol that we had to be able to do this in our own lab. So a clean room and trained personnel to be able to process and grow these cells, and we're able to then produce these lymphocytes to be used for therapy. But it's a heavy lift, and so it's not something every center could do, and what we are going to need to rely on then to make this broadly available are our partners in industry. And so there are, all my colors are totally messed up. Anyway, so there are now a number of industry partners who are developing commercialized ways of using this same technique to treat patients with adoptive immunotherapy. So these are the results of a recent trial that we heard the news about, and you're not going to be able to see any of the words. So I'll just tell you that this trial included patients who had failed previous standard therapies, the PD-1 drugs. 100% of the patients had had PD-1 drugs. Most of the patients had had the ipilimumab, CTLA-4 type drugs. Many of them had also had the BRAF and MEK inhibitors. So these are patients who had demonstrated an ability to grow through our standard therapies. And what they found was over a third of the patients had objective responses to TIL infusions. And actually the percentage of people that benefited is probably higher than that. As you'll see, it did still require the lymphodepletion, it did still require the interleukin-2, but the patients, oh, there's the words, but the patients did really well. And so just to show you these waterfall plot, and again, each of the bars represents a patient in this study, and if the bar goes up, their tumor grew, and if the bar goes down, the tumor shrank. And you can see the vast majority of them had their tumors get smaller with therapy. And then again, this flat curve. So the durability, they have not yet reached the halfway point in terms of people progressing who had responses. So how can we make it better? One problem we face is we can't always generate cells that identify and respond, at least in a way we can measure attacking tumor targets. And so can we program this to happen? And there are potential ways to do this, actually just using lymphocytes that are taken out of the peripheral blood. And they can be genetically modified so that they can identify and attack and destroy cancer cells. And there are a couple of ways this can be done. One is with modification of this T cell receptor, the steering wheel, right? You can insert into these cells your own steering wheel, your own navigation, and give them a receptor that is specific for a melanoma target. You can also have what's called a chimeric antigen receptor cell, a CAR T cell, where you've inserted a gene which is something more akin to an antibody that's on the surface of the cell rather than a T cell receptor, and converted into a weapon that way. And they have advantages and disadvantages, these two modified T cells. One nice thing about the T cell receptor strategy is you can identify things that are abnormal inside the cell, because the way our cells work is we present little things on the surface of our cell that tell the immune system what's going on the inside. And so if there are mutations that are only present on the inside of the cell, you can still react to them with this sort of technique. The CAR T cells react to things that are presented on the surface, so you have to have a surface target. And they've mainly been used now for hematologic malignancies, but are now standard therapies in hematologic malignancies. So the transgenic T cell receptors, there have been a few studies in melanoma of that, and this just shows the number of lymphocytes that infused after they are put in, and they can grow actually after they're put in. The numbers go up for a while and then come down again. And they did see in this study that many of the patients had indications of response on their x-rays. Their PET scans got cold, which indicated that they were having some sort of response. Unfortunately, it wasn't really durable for the patients in this study, and presumably because they just had a very small number of targets that they were going for, and maybe the tumors became resistant or got rid of those targets. But some hints that it may be effective. And then the CAR T, again, mainly used for lymphomas, leukemias, where this gene is inserted, and then they can see these indications of response. Their bone marrows then normalize with time, and this on flow cytometry that enables you to identify the cancer cells in blood. All the cancer cells which you see here go away after these cells are infused. It can get even more complicated, so you can take a tumor out, you can do whole exome sequencing, identify all the mutations that are expressed in the cell, predict what their mutations and the antigens that would come for that would be, and then find lymphocytes that react to those neoantigens and then give those cells back. So we can make it even more complicated, and there's great things to come. So new directions and open questions. Can we use TIL for other tumor types? And it looks like we probably can. Lung cancer, cervical cancer, maybe ovarian cancer, some breast cancer. So we'll see if it can be distributed to those. How much of this lymphodepletion do you really need? Can you do it in a more gentle way? How much IL-2 do you need? That's a really critical question in terms of opening this up to more patients. How do you pick the best lymphocytes? And there are a number of companies that are working on ways of making better TIL for use in the clinic. And how do we grow them? How do you make them better so that they're more effective once we give them back? So more to come. I want to thank my partner in crime. I've been together with him for a long time and look forward to many more years to come with him. And really critically important for all of this are all of the other people that are here, that are not giving talks here today. This is them in their normal work outfits. But these are all the folks at the Angeles Clinic that make the infusion work, that make radiology work, that make the nursing work, that coordinate all the appointments, all these things that have to happen to make all this possible. Those are the folks that make it happen. And to all of you who volunteer for clinical trials, you make this possible as well. So thank you all very much.
Did a surgeon just get up and give the best talk here? I cannot handle that. There is no truth in the fact that that person there was modeled after me. I was pudgier back then. But I will say that it's amazing to work with surgeons who know both ends of it and who can really relate it in a lay language where everyone, including myself, can understand. In LA, it's really difficult because I go to parties and I always say like, oh, I work with the best surgeons, they're great. And then I get a bunch of people around me and then they say, well, okay, well, what do? Do they do cosmetics or injectables? And then when I say no, they're gone. But truly, the ability to work with surgeons who understand all of it is phenomenal and important. What I couldn't do is allow Dr. Ferries to have the best talk or the best surgical talk here. So I brought in a ringer, and that's Dr. Ganji. Dr. Ganji joined us from Moffitt, where she trained classically in surgical oncology and melanoma surgery, and has been with us now 5 years. It seems like just yesterday, but is really an example of how surgical oncology has changed, not just for big surgeries or for early surgeries, but also to help us to get our patients from treatment to treatment to treatment in order to find the right therapies, and how we work at the Angeles Clinic for our trials along with our major partner, Cedars Sinai Medical Center. I will say Dr. Ferries put up MD Anderson and the NIH, and we're much smaller, but this is a model for every place throughout the United States. Those people that used to fly in from Nebraska can get this in Nebraska. Those people who used to come from Las Vegas can get this in Las Vegas and can stay at the Encore overnight. So it's available for everyone, even in a place like this that's small and compact, and about 30% of it is cafeteria. So with that, I'm going to invite Dr. Ganji to talk to us about surgical therapy in melanoma.
Thank you for the kind introduction. Dr. Hamid lied, and he actually brought me here so Dr. Ferries could look even better, but I'm not even sure that's possible. So my job is to talk to you about surgical therapy for melanoma, novel paradigms. I have no financial disclosures. I will say, in attempting to make this talk, the first version of the talk was that I wasn't talking about surgery at all, and that's probably the novel paradigm, is that we're doing less and less of it. So it's been revised really to focus on surgery. The other disclosure is that if Dr. Ferries considers himself a simple surgeon and not intelligent, then I have the intelligence of an earthworm. So with that, we'll kind of start broadly. Melanoma is known to be among the top 5 cancers diagnosed in the U.S. These are estimates from 2020, and you can see that in both males and females, there are still high rates of melanoma. The 2022 ACS incidence predictions were that we would have over 2,000 new cases of melanoma predicted for the year 2022, split fairly evenly between non-invasive, meaning not having the ability to spread to lymph nodes because they're fairly early, and invasive cases, and over 11,000 of those cases predicted for the state of California. So we're very fortunate to have people like Dr. Hamid, Dr. Mehmi, and Dr. Ferries here in our own backyard to treat these newly diagnosed patients. And really, it's the highest number of cases of any state, followed by Florida and Ohio. So again, my goal is to talk about the evolution of melanoma treatment from a surgical standpoint. So for that, we always require a little bit of history. And it was really very long ago that the first recorded of melanoma was predicted from the 5th century BC. And the word melanoma was derived from the Greek mela, black, and oma, tumor. And it really wasn't until 1787 where we had information about the first surgical removal of a recurrent melanoma of the jaw from a young male. So we did know about melanoma long ago. And in the late 1800s, it was Herbert Snow who first advocated for excision with prophylactic lymph node dissection, meaning we were doing very big surgeries and essentially removing huge chunks of tissue in addition with lymph nodes. And in the late 1960s, we finally devised a scale for determining thickness and what kind of surgery may be useful for patients. So we've gone from aggressive to less aggressive, but we'll talk about that a little bit more. So typically when I see a patient with melanoma in my office, my goal, and I fortunately work with very good dermatologists and physicians who refer to me, so a lot of this work has generally been done for me before I see the patient. But the question is, do patients have symptoms or distant disease? We do a physical exam looking at the lesion and surrounding pigmentation. This image here is typically not what I see, because usually this is what the dermatologist sees, and usually there's been a biopsy of this particular lesion. And that's why they end up in my office. But if this were to exist, then this is a suspicious lesion that should be biopsied for a variety of reasons. And then if they come to me with a pathology report, then I look at things like the thickness, concerning pathologic features, like is there ulceration or there significant mitotic figures, and then the subtype of melanoma, and that might determine whether they're eligible for surgery. And typically, there's no imaging that's necessarily done unless we know that the patient has bulky disease elsewhere. And again, typically those patients don't present to their surgeon first. They'll go to someone like Dr. Hamid or Dr. Mamie and our esteemed colleagues sitting here. So those patients will then be worked up appropriately with additional imaging if necessary. The reality is that most patients will actually present with localized disease. And what that means is disease is kind of limited to not yet having spread to lymph nodes or being distantly metastatic. And you can see that the 5-year relative survival rates for patients with localized disease is actually quite good, and it decreases as disease progression continues on. So when we're talking about localized versus non-localized disease, what does that mean? We typically we stage patients. So staging, you may have heard and you've been told you're a stage 1, 2, 3, or 4. And staging is based on 3 factors: the T stage, which is the tumor depth, the N stage, which is nodal disease, and then M stage, which is based on distant metastases. And these pictures here essentially just represent, depending on how deep the lesion is, that'll give us the determination of the T stage or the tumor stage. And if there are no lymph nodes or distant metastases, you're typically a stage 1 or 2. And this is an entire talk in and of itself, with modifications that were made in the most recent 8th edition. So I'm going to focus first on the management of early stage disease, which is really where the surgeon fits for now, and we have not yet become obsolete. And the management of melanoma for early stage disease is that the primary lesion, so that site that I showed you earlier that the dermatologist had biopsied and then sent to me, is excised, and it's excised widely, and I'll explain what that means in a minute. And given that even these early lesions have a predilection for nodal spread, depending on the depth of that initial tumor, you may or may not be told that you need something called a sentinel lymph node biopsy. But the reason that we worry about these nodes is because nodal status tends to be most prognostic for survival, and it determines whether you're eligible for additional therapies. And a majority of patients, like I said, will present with no clinical evidence of nodal metastases, meaning when we're palpating your neck or your underarms or your groins, depending on where your melanoma is, we don't actually feel any big lumps or bumps that are concerning, so that means you have no clinical evidence of nodal metastases. That doesn't mean that there may not be micrometastatic tumor or tumors that we just can't feel in those particular regions. And historically, as I mentioned, the recommendation was to perform lymph node dissections, meaning we were taking out big packets of lymph nodes. We were doing the same thing for breast cancer, and I'm sure you've seen, if you look back in medical textbooks or pictures, people who have swollen legs or arms because they've had these very extreme nodal dissections. Some of that has improved just with our surgical techniques, but the reality is that that's a possibility. So we were doing big surgeries and realizing that only about 12 to 20% of patients actually had evidence of metastases with early stage tumors. And so multiple studies have been done, and Dr. Morton, who's essentially the godfather of sentinel lymph node biopsy for melanoma, with whom Dr. Ferries worked very closely, and Dr. Ferries have modified the way we manage nodal disease and evaluation of nodal basins. So I'll go back to the nodal basin in a second, but with respect to wide excision of the primary, what that means is, again, you have the spot here on the top, and again, typically this has been biopsied, and depending on the depth on the biopsy, right, so we know if it's in situ, meaning early and lower preponderance from spreading into lymph nodes, thicker essentially as you go down. Depending on the thickness of the tumor or the T, the surgery involves an excision of this particular thing, and from the very edge we then measure out 0.5 to 1 or 1 to 2 centimeters, or an entirely 2 centimeter excision. So typically what I tell my patients, the reason you kind of see this eye is because skin doesn't tend to close up. If you cut out a circle, it doesn't heal nicely in a straight line, and so it typically requires that we create some kind of an oblique or moon-shaped incision in order to close that back up. But the reason for that is because there have been multiple studies that were done looking at how much adjacent skin we need to remove in order to get all of the tumor and limit the recurrence of disease, and that is why such a large chunk of tissue, adjacent healthy tissue is taken. Obviously, for this patient, that's much more significant. So if this patient presented with no clinically positive nodes, this is someone in whom what Dr. Patel talked about with neoadjuvant therapy, this is someone we may consider for that or other injectable therapies that I'll talk about in a moment to kind of minimize the extreme of surgery for having to remove that area. With respect to nodal status, I had mentioned Dr. Morton. So he was the first to report on the sentinel lymph node biopsy as a less invasive technique to evaluate nodal basin status. And essentially what that involves is you have your primary melanoma here. We inject some blue dye and some radio tracer, which allows us to count radioactive uptake, essentially, in the specific lymph node basin. Because you can imagine, if you have a melanoma, let's say smack dab in the middle of your back, right? So you have multiple nodal basins that are potentially draining that area of skin. It may go to both your underarms, it may go to both your groins. And if we treated this historically and said, okay, well, let's just cut out every potential lymph node basin that may have tumor within it, we're cutting multiple different sites for potentially limited benefit. So the sentinel node biopsy allows us to inject peritumorally, meaning where that tumor is, and then modern day, you go to X-ray essentially, and they're able to see where the uptake of that radio tracer is, and it might say, well, this particular spot in the middle of your back drains only to your left underarm or to your right groin, or potentially both. So it gives us some direction in terms of where we need to go, and then at the time of surgery, we inject some blue dye peritumorally, and in using essentially a counter which picks up that feedback, the radioactive feedback, and using our vision, it allows us to make a very small incision and pull out the first lymph node in kind of that echelon of the draining lymph node basin. And the thought there is if the tumor had spread from this primary site, it's most likely to go to this first node before it drains to any of the other ones. And so by removing this one that has the highest uptake and the most blue dye, it should give us a representation of if you have additional lymph nodes in that particular basin. And the MSLT-1 trial, which is one that Dr. Morton performed, essentially suggested that if patients had a positive sentinel node, that they benefited from having a completion lymph node dissection. But the reality in looking back at that data was it didn't really seem to improve overall survival significantly. And so then Dr. Ferries and colleagues further studied that. And now we feel that we can actually watch patients who have a positive sentinel node with ultrasounds, and we may suggest additional therapies. So again, the evidence-based guidelines for sentinel node biopsy are in patients who have very, very thin tumors. No sentinel lymph node biopsy is recommended, or melanoma in situ. And then in other patients, we essentially recommend sentinel lymph node biopsies. If you come in with clinically positive nodes, from a surgical standpoint, historically, we would recommend a complete lymph node dissection. Currently, given the number of therapies that we have available and how well they work, there are a slew of other options. So that's just something to consider, but since I'm a surgeon, I'm telling you what surgical guidance is. And then so standard nodal management based on these randomized trials, essentially, if you have a positive sentinel lymph node, and this is based on MSLT2 data, generally we'll follow those patients with close observation and ultrasound every 5 years. I'm sorry, for 5 years, every 4 months for 2 years, and then 6 months for 3 to 5 years. And if those patients have recurrences, we can certainly move forward with a completion lymph node dissection. For patients with clinically palpable lymph nodes that are limited to a particular lymph node basin, again, we recommend complete dissection for those subset of patients, and we may or may not offer adjuvant immunotherapy or targeted therapies for patients with nodal metastases. And Dr. Ferries, I agree with you, my colors are all messed up too. So for these early stage patients, what are new paradigms or novel paradigms? Well, certainly we're doing more and more minimally invasive surgery, which involves laparoscopic operations or robotic operations, particularly in the area of groin lymph node dissections. Both for superficial and deep lymph node dissections in the groin, we can perform these videoscopically. I didn't post actual pictures because typically patients don't always like to see that stuff. So here are cartoon images of how we may do that. We essentially put in some trocar sites. We can essentially put some CO2 in that area, which allows us to see better, and then you're dissecting on the same types of structures that you would be dissecting on open, but at the end, instead of having a large open incision, you essentially have a few small incisions and a drain, and the patients tolerate this significantly better. Similarly, for robotic surgery in the deep pelvic space, this is something that we can do. And then in the setting of patients who may qualify for resection of their metastatic disease based on limited progression of disease or really good responses, those are things that we can perform, again, less aggressively. So this is supposed to say what about recurrent or locally advanced disease, but the considerations essentially for those subset of patients are the location, the number of deposits, the previous excision to clean margins, whether the patient had received any previous treatment, and what comorbidities patients may have. So in advanced patients, we generally recommend a multidisciplinary discussion and shared decision-making between essentially medical oncology, surgical oncology, and the patient in order to determine what might be the next best treatment paradigm for you. With respect to local recurrence, so this is a patient who had a previous excision. You can see here, the surgeon did a nice job closing this in a straight line, but this was a local recurrence. And this patient had no additional disease or recurrent disease elsewhere, worked up. So in this patient, you can certainly recommend therapy, but we also have the option of just excising this particular area with 1 centimeter gross margins. And there have been studies that have been done retrospective in nature, meaning we look back at patients we have treated this way and found that, there are a significant number of patients who may have no further progression of disease. After this comes out, about 19% of patients will have no further progression. And at a median follow-up of 39 months, that subset of patients had no progression in about 77% of them. Some patients will develop recurrence. It may not necessarily be a local recurrence. It could be a distant recurrence or a regional recurrence, meaning to your particular nodal basin. And then some patients will have recurrence of distant disease. What do we do with sentinel nodes in those patients? There's really no good data to suggest that we should repeat sentinel node biopsy in those patients. So again, there's no increase in overall survival, but there may be a slight difference in disease-free survival in those subset of patients. What about patients who have essentially multifocal disease? So again, Dr. Ferry showed a similar picture in a patient who had been treated with BCG, but you could see there's multiple sites of recurrent disease in this chest, and then here in this leg, here on the scalp. So we have numerous intralesional and topical therapies. Dr. Ferry spoke about BCG. One that we use more commonly now is T-VEC or Telimogene laherparepvec or Imlygic, which is an injectable herpes virus, which essentially you can inject into a select number of tumors. Typically, we'll pick the largest ones in a patient who may present with this pattern of disease. And the thought is that the herpes virus can selectively replicate and lyse the tumor cells, and then it actually promotes release of GM-CSF, which can stimulate your immune system to essentially kill some of the adjacent tumors. And in a fairly large study that was well tolerated, there was an about 80% response rate with 50% complete responses and 30% partial responses with 40% durable effect. And then, I don't know why the text on the bottom isn't showing up, but these are topical therapies like Aldera that Dr. Ferries had also mentioned, which have fairly good efficacy, and you can see that this patient had essentially almost complete resolution of his disease. You can imagine that trying to surgically remove this would be quite aggressive, potentially with no benefit, and require a big free flap. Some other areas of treatment for someone who may have this pattern of disease in an extremity, especially the lower extremity, is hyperthermic isolated lymphoperfusion. This was described in the 1960s. This is pretty aggressive. It essentially requires that we cut down in the leg to access an artery and a vein, and then we put some percutaneous catheters in there, and we treat you with chemotherapy melphalan, which is a very, very old chemotherapeutic drug, which derives from nitrogen mustard and TNF in Europe, and then we heat essentially the leg off. There's a tourniquet that's placed in the upper edge. And the thing to take away from this is it's possible we do have some responses of this particular tumor, but if you're potentially given the option of immunotherapy or other therapy, you may prefer that to having your leg cut open with these catheters, and there are significant complications that can arise, including limb loss. As I mentioned, given that we're going to less aggressive types of therapies, from that we derived isolated limb infusion, or ILI, which is a less invasive version of the ILP. And essentially this involves us cannulating those vessels in the leg with small catheters. And again, it's hyperthermic, but not as hot as the ILP. There's still inflow. You cause some acidosis and hypoxia to that extremity. And again, we treat these patients with melphalan as well, with no TNF. In these patients, there was no limb loss and typically only grade 3 complications, which are still significant. So we usually grade complications from a scale of 1 to 4, 4 being most significant and obviously requiring multiple additional medical interventions. 3 typically requires additional support medically, but not necessarily a significant intervention. So the differences between the two essentially are the most important. I think I would focus on, you have fairly comparable response rates of 44 to 60%. With ILP, you can only typically perform it once. If the patient had bulky nodes, you could do a concomitant node dissection. But again, this is for a very, very, very select group of patients, and typically now not something that we do as a second line or third line therapy. It's really, we have so many other things that are potentially beneficial. You've seen this slide before, and the reason that surgery is becoming more and more obsolete is because we have all of this stuff. And Dr. Patel had talked to you about certainly neoadjuvant therapy and trials, and we're looking forward to the data from SWOG 1801 and Nadina in terms of determining what the best sequence of therapy is for this subset of patients. The question again that comes to mind is, what are the adjuvant treatments for high-risk patients? And then what are some combination adjuvant and metastatic treatments where we may be combining multimodal therapy, which may include surgery. And here, again, the therapies that we have currently available for us allow us to think more strongly about metastatectomy in areas that may be resistant to the therapies that we're providing. So this is a group of patients that were treated with checkpoint blockade. And again, you can see the overall survival for the whole group. In patients who underwent resection of their progressive lesions and were resected to no evidence of disease, the survival was obviously better. And so again, we have to kind of leave surgery as one of the options, but it may not necessarily be first line for advanced patients. In conclusion, the treatment of melanoma continues to progress with improvements in prognosis. There's many options for the treatment of locoregionally advanced and metastatic melanoma, including MIS options. Multidisciplinary tumor board and shared decision making is really important. And as surgeons, we need to be involved in the evolving treatments beyond surgery specifically. So I tried to make this as surgical as possible, but we are becoming obsolete. So with that, thank you again for having me, and hopefully that was slightly educational.
That was a great talk. So at this point, I want to make 2 points. One thing is that everyone always asks me during this, oh, are you going to continue to do this again? Are you going to run this next year? How are you going to change it? Who are you going to bring? And I'm not the right person to talk to about this. Really, I just come and take all of the accolades for it. The person you should be talking to, and the person I want to recognize, is Connie Doe in the back. Connie is responsible for all of this, plus my sanity, and I just can't thank you enough for continuing to stay and continuing to lead this effort. I think the patients should really know how much went into it. I also wanted to say that one of the greatest things, not just Dr. Ganji, that's come from our collaboration with Cedars Sinai is our ability to expand and bring basic science and lab into the Angeles Clinic. And Alex Boyko is sitting amongst you as a spy, Alex. And so we have a few basic scientists who've come on and joined us and hope to hopefully bring us into the future. We will also take a break right now for about 20 minutes. I think we'll start again at 5 after 11. We have coffee and bagels and everything out there. Whatever you don't eat, Connie has to take home, so please help us. And we'll see you back in 5 minutes. In 25 minutes to listen to Dr. Sullivan.
2, 3. 1, 2, 3. 1, 2, 3. Testing. 1, 1, 1, 1, 1, 2, 3. Testing. 1, 2, 3. Okay, this is the Q&A. So I'm gonna come over here and talk. I want to make sure that we don't get any feedback. 1, 2, 3. Testing audio. 1, 2, 3. 1, 2, 3. Okay. We should be good. 1, 2, 3. Testing. 1, 2, 3. 1, 2, 3. Testing. Okay. One, two, three. Okay. So I'm going to turn them off.
All right, well, we're back, and we're almost done. But with breakthroughs come questions. You know, have we solved the issues? What do we do with this therapy that's now not the first therapy? How do we combine them? What does BRAF mean? I mean, I just had a patient who's been my patient for a couple of years. We were talking, and he said he's doing well. He said, "What about BRAF? Do I have the BRAF mutation?" And I think we've talked about that initially. It was part of 100 different conversations, and then it sort of fell back to the wayside. And really, there's been a lot of change in how we deal with targeted agents in melanoma. When I have questions about targeted agents in melanoma, I go to some of the slides I've stolen from Dr. Sullivan over the years. The answers invariably are in those. I won't give him the satisfaction of a call, but the answers are there. And you know, we've talked about lineage and who trained what, and who did. Dr. Ferries has been trained by Don Morton, who was the dean of surgeons, and Dr. Ganji has come from Moffitt, where the surgeons reign supreme over the medical oncologists. And it's true that Dr. Sullivan has learned from one of the leaders in targeted therapy, and has taken all of that and just moved it another level higher. So I'm sure you will enjoy the talk. It's always a intelligent, entertaining discussion, as per the discussion last night. So please come on up, Ryan Sullivan.
Well, thank you so much. That was a very kind introduction. I hope I can live up to it. I'll do my best. Boy, it's been a long time since I've been up at a lecture, and I can't figure out which way to go. Okay, good. So this is my task, is to talk about BRAF-targeted therapy and other targeted agents, and thinking about today and also thinking about tomorrow. It's important to start with disclosures, and so these are some of my disclosures. I have one additional disclosure, which is we're living in an immunotherapy world, and I am an immunotherapy girl. I'm relatively certain Madonna never said that. Now, because immunotherapy is so dominant, one might wonder what I'm doing here. And in fact, Dr. Carvajal last night indeed asked me, "What are you doing here?" You're welcome. And seriously, I think a question that come up when you hear about these fabulous immunotherapies and truly the power to make someone's immune system better fight their cancer is the goal we should be building towards. It's the best chance that patients can have for durable benefit and our ultimate goal, which is cure. And so it's reasonable to ask questions like, okay, there's these other therapies. Is there any role for these after what we've heard from Dr. Carvajal and Dr. Ferries? And the answer is, I think there's a quick reminder here, or a few quick reminders. One is that there actually were two fundamental discoveries made in the last 30 years that have led to improved outcomes for our patients. The first, which is on my right, your left, is all of the stuff that people have talked about, immunotherapy. We've learned how the immune system interacts with cancer and how cancer interacts with the immune system, and we've developed and generated highly effective drugs for many patients. Not every patient, unfortunately, but for many patients. But the other thing we've learned, and that's on the right of this screen, is that cancer generally, melanoma specifically, is a disease of abnormal genes. And sometimes those abnormal genes activate, and those activations can lead to improvements for the cancer cells in survival, in growth, and spread, in evading the immune system. And if only we could target these mutations, we might be able to treat people better. And so this pathway, which is depicted on the right and on the left, what I've done is I've shaded half the patients who have what's called a BRAF mutation in red. I've shaded about the quarter of patients who have an NRAS or KRAS mutation in green. And I've shaded the about 10 or 15% of patients that have something called an NF1 mutation. And then over here, here's RAS. So this is like cancer signaling, actually general cell signaling 101 or maybe like 302. But ultimately, there's signals that get that kind of come from the outside world and interact with cell surface molecules that then activate stuff. And this is what can activate is this pathway that's also called the RAS, RAF, MEK, ERK pathway, or MAP kinase pathway. And in red are mutations in genes that we sometimes see in melanoma, so BRAF, NRAS, c-KIT, GNAQ, GNA11. You'll hear more about those later. There are other pathways that kind of can be parallel to this and interact. And we've now developed drugs that can block these proteins relatively well. And so for the remainder of the talk, I'll be discussing what we do when we block these proteins and how the blocking of those proteins may be optimally used for patients with BRAF mutated melanoma or some of these other mutations. And so in addition to the sort of things we've learned, so we have multiple adjuvant, I guess I even put multiple adjuvants in that sentence, which is interesting. So we have multiple adjuvant therapies for patients with stage 3 melanoma. So we heard about the Checkmate 238 study and the Keynote 054 study from Dr. Carvajal earlier. We didn't hear about this trial called the COMBI-AD, but what adjuvant therapy, which was described, is treatment that's given after surgery, so curative intent surgery, but there's a risk that the cancer will come back, and we give treatment to try and reduce that risk. So we have three options, and these three options, two of them are basically the same. Sorry to anybody who works for BMS or Merck, but these are drugs that essentially work the same. They're PD-1 blocking drugs. They look very similar. Dabrafenib and trametinib are two different drugs. These are BRAF and MEK inhibitors that are targeting that pathway that I showed earlier. The other thing that we also are going to talk about is there are many unmet needs in advanced melanoma, and these include what the heck do we do for patients when we've used up all of our immunotherapies and patients still have melanoma? Like, how do we treat those patients? How can we help those patients? We might be able to think up and dream up more immunotherapy combinations or give tumor-infiltrating lymphocyte therapy for patients. But ultimately, if that patient has a BRAF mutation, we're also going to be thinking about how to block that pathway. And if they have other mutations, we may also be thinking about how to block that pathway. So one of the things that I'm not sure Dr. Hamid steals these slides because he always makes fun of my stick figure patients. But this is my stick figure patient. I know that patients are real and they're three-dimensional, and I care very deeply for my patients. But for purposes of talk, I make them stick figures, so I apologize. So this is just a patient who has a BRAF mutation. They have completed their surgery, and they're sitting in front of myself or another medical oncologist, and we have a dilemma that we have to solve. What do we best recommend to this patient? Do I offer this patient BRAF-targeted therapy? Do I offer that patient immune-targeted therapy? And I'm going to briefly go through this, but ultimately in blue is BRAF-MEK combination, in red is double placebo, and this is relapse rates. And so if you're still on the curve, you haven't relapsed, and you can see that blue, there's more people who haven't relapsed than red, and this is with 5 years of follow-up. There's a lot of words on the slide. It works. It's FDA approved. It's an effective therapy. This is a summary in a different way that Dr. Carvajal showed of the two studies of the PD-1 blocking drugs. This is the Keynote 054, so that's pembrolizumab. This is the Checkmate 238, that's nivolumab. Again, the blue curves are higher than the red curves. That means you're less likely to relapse if you get the drug pembro or nivo than you are if you get the other stuff, which here it's placebo and here it's ipilimumab. So again, a lot of words. These drugs work. And in fact, when you do this thing that statisticians vomit over, but we do all the time, which is to cross-trial compare, it looks about the same. The 4-year relapse-free rates are about the same. The prevention of distant metastases is about the same. These are just different ways of doing it. This is a hazard ratio, but these hazard ratios are close enough, and this is comparing against an active comparator, so it's probably, if you kind of back of the envelope calculate, it's about the same. So I would just say that this slide, lots of numbers, but these drugs are basically the same in terms of how effective they are. So then what's the difference? Well, they have different side effects. BRAF targeted therapy is actually fairly toxic. About 60-70% of patients will have a severe side effect at some point during the course of their treatment. These other drugs, ipilimumab is way more toxic than placebo. It's more toxic than PD-1 blocking drugs. Pembro is a little bit more toxic than PD-1 blocking drugs. So you can look at severity and frequency, but you can also look at permanence. And I think what's important to note here is that there are permanent toxicities that happen with our immunotherapies that just don't happen with our BRAF targeted therapies. BRAF inhibitors work like every drug, or almost every drug, because I'm going to tell you drugs that they don't work like. You take the drug, a level in your body gets in there, your body clears the drug. If you're having a symptom or a side effect from the drug, it's there when you're at the peak dose of it, and when it clears out of your system, it goes away. So BRAF targeted therapy works basically like every other drug. Immune therapy does something very different. Now, it's the reason it's so exciting is that it can change your immune system so that you now have immune elements that are attacking yourself. And that may persist unless you actively reverse it. And so that's a distinguishing feature between these two types of therapies. And just more on chronicity, we call these side effects of immunotherapy immune-related adverse events. It's basically just immunotherapy side effects. And what we've shown in a large group of, I think I guess it's on there, 8 academic centers in the US and Australia, that about a quarter of patients have severe side effects, or about 20% rather. And of those, about 40+% have persistent side effects. So that we define persistence as you stop, and 3 months later you still have side effects. And the most common side effects are like thyroid inflammation and joint inflammation or achiness. But we see other stuff too. We see skin side effects. We see colon side effects. We see eye side effects and nerve side effects and lung side effects. So we can see permanent side effects with these types of drugs. And so kind of putting it all together when I'm in front of my three-dimensional real patient, but for the purposes of this, the stick figure patient, BRAF targeted therapy actually probably works better when you have less disease. Also, like other types of treatment in cancer, when you can't see it, there's less tumor, you actually don't need to kill as many cells. These drugs can probably kill many of the cells that are still there and help prevent it from coming back. BRAF and anti-PD-1 therapy are similar in how well they work in the adjuvant setting. And I think that another important point about PD-1 blocking drugs is the immune system may actually be interesting in that in the setting of these drugs, it may not actually matter how much tumor you have. If you have a lot of tumor, it probably matters. But if you have a little bit of tumor that you can't see on a scan or a little bit of tumor that you can see on a scan, I'm not convinced that these drugs work any better in the person who has a scan that doesn't show any and just had surgery, or a patient who has a relatively small amount of tumor on a scan, because the immune system sort of works very similarly in that setting. It's hard to imagine it's going to be dramatically different. And the last point is that there's more severe side effects with the BRAF targeted therapy, but there's more chronic side effects with the immunotherapy. And so what do I do when I'm faced with this? My bias, although these are long conversations that I have with patients, and we go through all of these features, but my bias is to give the BRAF-MEK combo because it's potentially curative at a similar level as the PD-1 blocking drugs at that time. There are reversible side effects. And salvage, which is essentially, can we cure somebody later? Salvage immunotherapy seems to be effective in this patient population for some patients, but at a rate that we might expect that they would have long-term control if they didn't receive this treatment. So that's one potential way we can use BRAF-targeted therapy in this era. Back to here, I think one important thing to say in the metastatic setting after immunotherapy has stopped working, I'll get to that one important thing to say, but here's my version of the slide you've seen. In red are the BRAF targeted therapies, and in the sort of darker blue or black are the immune therapies. Lots of progress, but now we have this patient with advanced BRAF mutant melanoma, and Dr. Carvajal showed this already, but I think it's important to know it's not either or. We're just going to give patients probably both at some point in their case. The frontline data with BRAF targeted therapy demonstrates that it works, but this last one is the study that Dr. Carvajal showed, that essentially patients who start with immunotherapy, in this case ipilimumab-nivolumab combination, ultimately do better than patients who start with BRAF and MEK inhibitor therapy. But we have limited data about how well these drugs work after immunotherapy. With that said, I think there is a role for every patient who has metastatic BRAF mutant melanoma, with rare cases excluded, who are not cured or do not have long-term benefit from immunotherapy to receive this. Said another way, I don't think a patient should probably go on hospice if they have a BRAF mutation, if they haven't received BRAF targeted therapy. We can improve their life, we can extend their life in that scenario. Here's the only significant data we have in a prospective study about how well BRAF targeted therapy works in the frontline setting. So Dr. Carvajal showed you this, and in the second line setting. So essentially a very similar response rate. 5 minutes, thank you. We don't know for how long this worked for those patients, but we think it's similarly effective in both of those settings. And so we think that this is something that a patient with BRAF mutant melanoma after immunotherapy, if they haven't gone into remission, should be considered for. Now, it is sort of becoming an existential question, which is, can we make BRAF targeted therapy better? Is there anything we can do to modify this complicated mess to try and make the BRAF inhibitors better? And so, again, lots of boxes and ovals and things like that, but ultimately, every darkish thing here is potentially targetable with drugs. And the one thing I'm going to talk very briefly about, and so I've started, all of these things are potential targets that if we could safely give those drugs that target those things with BRAF targeted therapy, we'd probably make it better. It's a little bit more complex though, because if you start adding a bunch of these drugs, you start increasing the side effects. But one thing I'm going to talk about very briefly is this concept of apoptosis and targeting it. Apoptosis is otherwise known as programmed cell death. It is a way that cells can die. It's a way that cancer cells can be killed. And it's also a target because cancer cells are actually really good at preventing this from happening. And so there's a drug that's actually used, or a drug actually, a second generation of that drug is used in some certain types of leukemias, lymphomas, that blocks a key mediator it blocks a key blocker of apoptosis. Navitoclax is sort of like that. And so we added navitoclax to dabrafenib and trametinib. We did a phase 1 study, fairly complex. We had a bunch of dose levels. And the point of the phase 1 study was to find a dose. And in red, this is the dose we found. Importantly, the D, which is dabrafenib, and the T, which is trametinib, this is the same dose that we give to patients if we were just going to prescribe it to a BRAF mutated patient. So we didn't have to lower the dose of the drug that we know, or the drugs that we know are effective in this patient population. And we added this drug, navitoclax, at a dose that's a little bit lower than the maximum tolerated dose, but we felt pretty comfortable with this result. Well, I'm not going to try and read that for sure. The second part of this study was actually a randomized trial. And we randomized patients to receive 3 drugs or 2 drugs. We just finished accrual about 2 months ago, and we'll probably do the analysis of the comparison in about 2 or 3 months. If this is a positive study, it'll be really exciting, and it may help to open up a new avenue in terms of how to treat patients. Importantly, most of these patients, even though you didn't have to, but most of these patients receive prior immunotherapy, right? So when we initially launched this, which I'm embarrassed to admit was in 2013, we didn't have PD-1 blockers that were FDA approved. So a lot of patients actually could have conceivably gotten this as their first therapy. It's not something that we've been doing in the last several years. So this will actually be appropriate. The data we get will be appropriate for this era. What about BRAF MEK inhibitor resistant patients? What about NRAS mutant patients? What about NF1 mutant patients? Well, I'm going to flip off of this screen very quickly, and then you're going to have to remember everything, and there's a test. But this is actually a figure from a paper that we've just published, and Poulikakos is a leader in bench research about this pathway. And he and one of his postdocs came up with this figure. But ultimately, what it shows is there are a lot of targets for cancer cells that activate this pathway. And importantly, what we're doing with this information is we're using things that target. So we're targeting ERK. We're targeting RAF in a different way. We can target MEK. We can target RAF in a different way and MEK. And what we're seeing, and you've seen waterfall plots now, these are patients who are very resistant to treatment, and a lot of these patients are having benefit. And so we're starting to see some evidence of activity, and what we're hopeful that we'll see over the next few months, next few years, is that these combinations begin to get into larger patient populations, and that ultimately we might be able to translate some of these into FDA-approved regimens. This is a combination that actually showed pretty nice effects. And what this has done, even though it does not that many patients and it's like only half of patients did well, but what it's done is it's led to a number of new combinations that are making their way into the clinic with different drugs that are slightly different in terms of how toxic they are, how tolerable they are, what they're targeting, and I think at least 1 or 2 of these will turn out to be quite effective for our patients. So to conclude, I think first of all, there's a role for targeted therapy early in the adjuvant setting and late in patients with metastatic disease after immunotherapy for patients with BRAF mutant melanoma. So despite the fact that we are living in an immunotherapy world, there is still a place for targeted therapy. And there are new targeted therapy strategies that are emerging to treat BRAF, NRAS, and other oncogene populations or driven populations. And hopefully over the coming months and years, we'll have more data to share on that. So with that, I thank you, and I look forward to the question and answers later.
Thank you. Ryan, you know that the Angeles Clinic stands alone in West LA, but we're surrounded by a lot of plastic surgery places, and a couple of those patients got onto the feed, the live feed, by mistake. They called me. They said, I want to look like Dr. Sullivan's stick figures. So we booked them with you, and they're going to get Botox and a facelift with Dr. Ferries. But a great talk. Fantastic. Thank you. So I tasked myself with talking about the future therapies and newer targets. One, because there's never been a greater time to look at what's available. Two, because we're taking information from other solid tumors and bringing it back to melanoma, whereas in the past it was all the work being done in melanoma and sent to other tumors. And three, I can say whatever I want, and no one's going to call me on it. But it's really nice. So I titled it WTF, and that is, what's the future? Here are my financial interests and relationships and conflicts. But two things. One, the more conflicted, the better and more even keel the talk. And number two, if you look around, there are people from multiple pharmaceutical companies here just that should make you feel happy in that we do have a lot of basic science partners, surgical oncology partners, leadership in the hospital partners, but also now pharmaceutical partners who are really invested in the well-being of our patients and have a whole host of options. And this was not the same when we started 13 years ago talking about an AIM symposium. Here are the approved agents and what we have, and we've really moved average survival from 6 months to 6.5 years and more. And I feel, so what are tomorrow's questions that are due today? This is really what's going on. What do we do after the LAG-3, the relatlimab approval? What happens with CTLA-4? And then when you've exhausted Dr. Sullivan's options, what about TIL therapy? Are we done? Is it, what about brain metastases and leptomeningeal disease? We're seeing less and less patients have those issues, but when we see them now, they've exhausted all the options we've had. And then, of course, the whole reason we do this is the belief and buy-in into clinical trials, clinical trials, clinical trials. Dr. Sullivan talked about a trial that was started in 2013 and is reading out now. I would say that when we started with our initial immune checkpoint trials, it slowly seeped into the community. It took 10 years for people to even understand that we were doing trials with CTLA-4, but now you can start a trial and finish it and report it in a couple of years, 2 years. In the time that the patient who gets on the trial, 2 years in, there's data to help the whole cohort of people around. So it's fast, it's moving quick, but we need more answers, and it's not easy. This was what was, Dr. Ferries has shown you this, but this is Jed Wolchuk's initial T-cell, blah, 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 blah. But in LA, as we saw driving around, you need a better vehicle, and you need to do better. This is a electric car with lights and bells and whistles, and that's what I like. We're also learning that not only are we doing more, but we have a better goal. Our initial goals were, we just need to stop this thing in its tracks. And now we're getting people to this thing called CR, complete response, where everything is gone, or complete metabolic response, where you see things on the scan, but on the PET scan, they're not active. They're not metabolically active. And what these curves are telling us is that we need to work and build and do more and add more to get a greater proportion of patients to that CR, because those are the ones that are going to do the best long term. So we can't just sit on our laurels. And so I went to AACR, the slides at AACR, which just happened in New Orleans. unlike some of my colleagues who were, going down Mardi Gras and gathering beads, I looked at these things, and I think it's really worth mentioning that the ability to think beyond just one drug and another drug, and let's just give them together, is where we're going. Let's take one drug and make it like 5 drugs. And so this is a monovalent bispecific antibody. All it means is instead of just having PD-1, it has PD-1 and CTLA-4. And that allows it to target better. That allows us to get higher doses into the right places and less toxicity. So that's what this is saying. You can get faster inhibition, less toxicity. And that's been the problem with CTLA-4, the ipilimumab and the Yervoy you hear about. When you combine them, there's too much toxicity. But this type of bispecific drug can give you more. And then you can flip out and give LAG-3 instead of the CTLA-4. And these trials are ongoing in multiple solid tumors. This trial was a MedImmune drug, was in many, many cancers, and they saw responses in gastric, bladder, Merkel, cervical, and others. So these are coming, and they're coming for patients who have seen the standards in melanoma and need other options. And so many years ago when I came to Angeles and talked about expanding the clinical trials department, I went in my office and there was no expansion to be had. There was nothing to buy. There weren't these trials, but now it's the exact opposite. We are looking and teasing out of tens of trials offered to find the best trials out there. Again, we're also looking at giving drug directly into tumor to try and avoid side effects, but to get it to start that tumor death that can then work systemically with the other drugs we give. And this is a CTLA-4 that's injected directly into the tumor, and you can see that it works distantly and locally. And this is in metastatic breast cancer. That's why I tell everyone, to come to the melanoma talks, because you can take this back to other solid tumors and make breakthroughs. We have a lot of checkpoint inhibitors and checkpoint activators that are going into multiple clinical trials moving forward. And we have a lot to do here to understand the tumor microenvironment. But I'm just going to talk about one thing here, and these are adenosine inhibitors, partly because we have a trial of it here at Angeles. And the idea here is one that's super interesting. And no, we missed one. But it's that ATP is needed to give energy to our T cells to activate, go after the tumors, and to survive, to inhibit the bad T cells that put the brakes on your immune activity, to get the other immune cells into the area of the tumor, etc. And we know that this ATP adenosine axis is important, that when ATP is used up and becomes AMP, so ATP, you need it for your cells to grow and have energy, but the tumor preferentially takes ATP and breaks it down to AMP and then adenosine. And adenosine is inhibitory to our T cells. What we've found is that we can block CD73, which is an enzyme there, and we can block the target of the adenosine, and we can also block this other CD39 that changes ATP to AMP. Guess what? We're putting them all together in one trial. And that's the future. Just not checkpoints, but other areas. When I hear people talk about neoadjuvant therapy, I think about all those patients who then come and say, I can't do that. I don't have tumor in place. But there's great stuff, great work that's coming out of multiple companies, including Moderna. You're all familiar with Moderna. And they're making vaccines off of the tumor that's already been pulled out of the body. And in those patients who are getting adjuvant therapy without tumor in place, they make vaccines that express those proteins that can increase the immune activity. If you think back to the beginning of this meeting and Dr. Patel showing that increased T cell activity when the tumor is in place, you don't just need to have the tumor in place. You can have the tumor made into a vaccine given along with it. And guess what? That trial is already done, and that data will be presented shortly. We took a bunch of patients, a bunch being 150, and we gave 100 of them the adjuvant PD-1 inhibitor plus a vaccine made out of their tumor, and we gave 50 of them just the adjuvant PD-1 checkpoint inhibitor, and we were blinded to who got what. And that data is now 2 years in, and will be ready to be presented shortly at a meeting by the end of the year. But you can identify these proteins that are the neoantigens, you can create a synthetic vaccine, and you can give it in combination with adjuvant and checkpoint blockade. You can also do it in the metastatic setting, and we've been doing those things, and that's coming forward. We then took a trip with Dr. Ferries and talked about TIL therapy. And that is, again, where the tumor tissue is procured. I don't have stick figures, but I have something between yours and Ferries' complex person. And so you take the tumor, you make these, you send it centrally, and they make these TILs and they send it back. Well, the idea is that we can do more here. And what can we do more? This is from a company that we're working with, and they are, I can't explain CRISPR, but CRISPR is a hot word. You say CRISPR and people think, this doctor knows what he's talking about. He's at the tip of the spear in treatment. But so CRISPR and CRISPR and CRISPR, what they found is that they looked at these targets that lead to immune dysfunction, and they can CRISPR or cut out that part of it. So the place that did this, Iovance, is CRISPRing out PD-1, but this company is CRISPRing out SOC-S1. And this gene is inactivated, and you have an engineered TIL. So you have a heat-seeking missile that does not have a problem with it. It's stronger, and it goes forward. And what this says is, look, it does better than normal TIL. So you get no cells. This is how your tumor grows, this black thing here. You get normal TIL. This is how your tumor grows. And you get this drug. This is in mice models. And you get this. So when people say 10-fold potency, you want to put that in clinical trials, and it's coming. What about changing some of the drugs that give toxicity? How about giving away in order to avoid some of the toxicities of TIL? So Dr. Ferries talked about interleukin-2 and how alone it can get rid of tumor, and how as an adjuvant, after you get those T cells infused back into your body, we'll go backwards here. You get those T cells infused, and then you get IL-2 in the hospital. How can we fix that? How can we make it better? And this is a drug that comes from a company named Obsidian, and they're doing their phase 1 trial at MD Anderson. And so they're going to start doing these trials at MD Anderson. They have, instead of systemically given IL-2, they're giving endogenous IL-15. That's another interleukin. We believe that it may be better, that it does not stimulate those Tregs, those cells that regulate your immune activity, and that you can see less systemic therapy from it. Anyway, there's a linker here, and you take a pill, and it activates that release of IL-15. So if you have someone, you don't have to give them these infusions. If they get toxicity, you stop giving them that pill that releases this. And you don't have to pay for the IL-2. There's no financial toxicity of it. So it's more and more and more. And here are all of those scientific slides that say, hey, it may work better. Improved TIL product demonstrating anti-tumor potency without the need for IL-2. I'm up for that. Sign me up for that. And honestly, I came in second for number of slides per minute of talk. So I've tried to cut those out. But what I've cut out from this talk is the fact that TIL therapy is not just for melanoma anymore. There's great data in cervical cancer, and it will be in front of the FDA for approval for cervical cancer. There's great data in patients with lung cancer. There are trials going on with head and neck cancer, and it's moving earlier and earlier and earlier, and these TILs are going to come to patients and are already in clinical trials of patients who show and say, I have metastatic melanoma. I don't know if I should get PD-1 or should I get TIL? And you go, guess what? You get both of those things. So that's upcoming now too. Why is it so interesting for us, and why are we pivoting our research program to try and bring some of these cellular therapies forward? It's not just because of this waterfall plot that Dr. Ferries adeptly educated us about. It's also about these things. It's plots. Thank you. You have to learn about these plots. Way back when I was a fellow, you didn't need to do plots. I mean, nothing really worked. So they said, it worked this, it had this response rate, it only worked for 2 months. So if they did like a swimmer's plot, it would stop like right there. You wouldn't need anything. But now you need to learn about the waterfall plots, that's how much shrinks. The swimmer's plots, very descriptive, that how long it works. And this is a very convincing response in patients who had seen, all of them, 100% had seen PD-1, 80% had seen anti-CTLA-4, which is Yervoy ipilimumab in one way or another. All of the BRAF mutant patients that Dr. Sullivan talked about had already seen BRAF inhibitors. And this worked. And it worked early. You see these blue diamonds? That's when the first imaging, a majority of them had already shown shrinkage. But here, 80% of the people that responded, their initial response to PD-1 therapy, which is the backbone, which is the standard therapy for every patient with metastatic melanoma who shows up in the door, 80% of these people, their best response to PD-1 therapy was no response. So that's taking a subgroup of our patients and telling them, we have something for you. If at 3 months when we look in, it didn't work for you, well, we have something for you. Next. Dr. Memi is going to come and talk about uveal melanoma. And he's going to talk to you about how immunotherapy is now working in tumors that we never thought it could work. But this drug, you'll hear a lot more about. I'm going to say it's worked extremely well. I call it a matchmaker. Those of you who are Jewish like me, it's a yenta drug. It brings these two things together, but it's like a bizarro yenta. It puts you together and wants you to get a divorce. It brings the T cell and the cancer cell together and hopes that you get a huge immune reaction. And we're doing a whole host of trials with drugs like this that take other solid tumors like endometrial cancer and ovarian cancer, those that don't respond to immunotherapy, and are seeing some promising results, right? During the pandemic, I learned to do a lot of animation on these slides. But this is the data of it of this drug in combination with PD-1 and CTLA-4 in cutaneous melanoma. So we are seeing about a 37% control of disease, and this trial is going forward. Guess what? It's being given subcutaneously, under the skin. You don't even have to sit somewhere. You just come in, you get a subcutaneous injection of this drug, we watch you for an hour, we kick you out. So it's great. The third type of plot you need to know is spider plot. So new things in your vocabulary is waterfall, swimmer, and spider plots. And this shows if you want the spider's legs to go downward and die like this, durable response. This shows patients who've had the standard therapies beginning to benefit. And we've taken patients off of this 2 years in and seen that they continue to have response. And that's the benefit of immune therapy. You don't have to continuously get it. Your immune system is active and going forward. Dr. Memi has brought forth other trials where we are working to utilize your own T cells, but we don't need to have you, come into the hospital, and we don't have to manipulate the T cells in a major way. The T cells come out, they're bathed in a jacuzzi of activators, and then they're given back to you in the clinic. So that's a wonderful future. You've heard about CAR T, but more and more this will come forth in other solid tumors, and then come into melanoma. And this is the flipped paradigm. It didn't start in melanoma, but it's coming in melanoma. And then, I'm going to run out of time. But there are other types. And this word is a new word for you too, allogenic. That means it comes from someone else. And all of the CAR T stuff we talked about is auto. It comes from you, the patient. Allogenic is off-the-shelf T-cell therapy. And that's NK cells that you can get off the shelf and give it to patients with other solid tumors. This stuff comes from Rizvani and others. Who is at MD Anderson. She's my favorite person at MD Anderson. Why? Because she's Iranian like me. And she's doing things like, we come to work and we're like, we're doing something great. So she's doing great stuff. She's taking cord blood, not even from an adult, cord blood, and utilizing it to make these NK cells to give to patients and manipulating those cells. So this is like a maestro. This was presented from MD Anderson Cancer Center. It's from her lab. And this is cord blood derived NK cells that have an innate engager. What does that mean? You don't need to remember that. It's basically a bispecific already hooked up to the NK cell. Oh, that's super. This was at AACR, which just could jaw drop moment, because you don't give them you don't give one and give the other. This together, one drug that takes the NK cell directly to where you want it, to the tumor. And you can see this happening. We have that bispecific in melanoma. We have TIL. We can do NK and go from there. What about brain mets? And this, I think, is towards the end. TIL therapy here. These slides I stole from Alison Betoff-Warner. This is data from the Iovance lifilocell that you saw in metastatic brain metastases, and she's going to be opening a trial like this. So we're not just sitting on our laurels saying we have success, but we're tackling some of the difficult questions. Here's a patient who had systemic response and also brain mets response. Damn, I'm like an ad for MD Anderson today. Please tell him to send a check. Third floor. But, Isabella Glitza is phenomenal, and the whole MD Anderson history goes back to looking at leptomeningeal disease. That's the last bastion of the worst outcome patients. There's a study Dr. Sullivan has of ipi-nivo and leptomeningeal disease. We know it works, but what if you've already seen it? This is intrathecal, giving it directly into the fluid in the brain. You don't go through the vein. You go, I'm going to put it in your brain. It's going to work, and it works. But there's better. This was presented from the Harvard group. It's not in melanoma. It's in low-grade gliomas, brain tumors in children. And they gave this drug, so this is CAR T. That's where Dr. Ferry said you change it, you give it something that hones the T cell to the tumor. This is CAR T, And they give it intravenously, that's IV, and they give it optionally into the cerebral ventricles, into the brain, a T cell. Just I'm going to put a needle into your brain, and da-da-da-da-da. And what they found was amazing. These are children who had neurological compromise, who had resolution of all of their neurological symptoms. What they found is giving it intracerebral ventricularly without lymphodepletion, not needing to give that systemic chemo to get rid of the T cells, but just giving it into the brain. You saw activation of the immune system, and this was unbelievable. That you're getting, this is your spine, this is the tumor. You don't want to see that. That's not great. But then shrinkage and shrinkage and shrinkage to near resolution. And the reason I use this pointer is because I want to bring your attention now to this. 2 months, 5 months, 9 months in to something that would have a very poor 1 to 2 month prognosis, if at best. So that's where we're going. And the next thing is to say that we are having better ideas of using imaging. So behind the cafeteria, we have MRIs, we have a PET scanner, and we need to learn how to use them. And this is an imaging tracer where we give you give it IV, and it hones directly to where the T cells are. So instead of saying, hey, you need to spend 3 months on this therapy, you can say within 8 weeks, we can do one early, see where your T cells are, and at 8 weeks we can see if we're getting those T cells to your tumor. This is uptake in areas where there is tumor, and it's lighting up showing that the T cells are there. We overlay them, we say success. We overlay them, and they're not on top of each other. We say, you need to go to something else. You don't need to come in another month or 2 months or 3 months. So with that, I am done. And I want to thank you for listening. I may have gone over, maybe a little much, but I will now introduce Dr. Memi, who will talk about ocular melanoma, and then we will go into the questions. Now, I can't do a long introduction for Dr. Memi, but to say that he's like a savior for us, for our program, and it's the greatest thing to come in and have a younger, enthusiastic investigator that calls you on, questions, what's going on, and leads you in the right direction. I need you outside of the clinic too to help, but Dr. Memi.
Wow, I don't know what to say after that. Thank you for giving me the opportunity to talk about one of the diseases that still is challenging. All the things you've heard so far, immunotherapy, targeted therapy, it's applicable to most of the diseases, and to some extent, we'll see how well we can apply it to too many phones in my pocket. Apply it to ocular melanoma. So I want to start ocular melanoma talk with a little bit of background. So these are my objectives for today's talk. I don't have any disclosures. Oh, stuff is missing. Anyway, so these are some of the sites where you can develop ocular melanoma. We won't spend too much time here. And most of them, they behave similarly in their prognosis and outcomes. I wanted to also give you a little bit of background in how common this disease is. It's not something that we see all that often. It's a rare disease, even though this is the most common primary cancer of the eye in the adult population. It represents only about 5% of all melanoma, cutaneous, mucosal are the sort of more predominant ones. We're seeing only about 2,000 new cases per year, and globally it's about 8,000. And I think the global numbers are probably a little misleading because they may not have the resources for diagnoses. So it's not that we're leading in ocular melanoma. I think it's just we're able to diagnose a little bit better. Some of the risk factors for this particular disease, they could be genetic or inherent in you. And they don't parallel cutaneous melanoma as much, though. As far as the outcomes from this severe disease, almost half of the patients go on to develop metastatic disease. A lot of them will have primarily in the liver, and that gives us some opportunities for therapeutic approach for those patients. And additional patients will have extrahepatic or outside the liver disease, and some will have combination of it. And that also presents challenges and opportunities for therapeutic options. Unfortunately, for this rare disease, we haven't been able to do a whole lot over the past however many years, even though we've made strides in leukemias, lymphomas, childhood diseases, malignancies, cutaneous melanoma, and breast cancers. Unfortunately, the fact that one, it's a rare disease, two, its behavior is quite aggressive. It presents a lot of challenges for improvement in treatments and improvement in outcomes. And we'll talk a little bit about target therapy as well. And also I wanted to say I'm talking in front of world's two famous uveal melanoma experts, and don't judge me, please. One of the things I wanted to highlight, but we won't spend too much time on this detailed slide, is staging that kind of helps us prognosticate how a patient will do based on what their primary disease is. And there are two sort of well-known methods. One is based on the size of the tumor itself, and the second is inherent genetics within that tumor. And it seems like when you are looking at the genetics, the sort of the inherent biology of the tumor, we're able to better prognosticate than just simple from this is how big the tumor is and this is the risk of it coming back. I think one of my old attendings used to say that sometimes the bigger tumors are not as scary as the tiny ones that you can't see, because they hold the nasty gene mutations that make it a lot more aggressive. So size isn't always the key. So gene expression profiling is also what I was talking about. So when you look at these early stage melanoma, and there's I think about 20 different genes, you look at them and they get split into two groups, class 1 and class 2, and then you look at their prognosis, how they've done. The class 1s tend to do well. That's the flat line up top, and they have low risk of recurrences, but class 2 do not, and they tend to have relapses early. Their disease is aggressive. And then on the long-term follow-up, you'll see that even there's subset of class 1 patients of uveal melanoma that have sort of more poor outcome compared to some of the other ones. And again, so then there's additional added on antigens on these markers on the cell surface or within this cell. It's called the PRAME antigen. And when you add that as a layer on trying to prognose the patient, that also gives you another step in saying, okay, these are the class 1 patients. If they're PRAME positive or PRAME negative, how will they do? What are their recurrence risks? And how can we watch them closely so we can detect their metastases or their relapses early so we can manage them better? So PRAME gives you another layer of complexity and helps you manage these patients better. And this is just to drive the point home that when you're looking at the genetic nature of the uveal melanoma, rather than the size of the tumor itself, you're better able to prognosticate. So now moving from once you've diagnosed the patient, you've given them some sort of prognosis, how do we go about treating the tumor in the eye itself? And there are a number of different options, and my ophthalmological friends are very good at explaining what they do, how they do it, how they follow you over the long term. And there are obviously adverse events related to these therapies, loss of vision or changes in vision, sort of irregular development of the blood vessels post-treatment. So there are managements for that. So again, quite complex of a treatment option, but I put you in a simple list here. So now we're looking at a patient who has been treated with or had their primary tumor treated, How can, and we've given them a prognosis. Okay, you know, patient X has come in, they've had a serious tumor, it's class 2, PRAME positive, and it's invariably is going to recur. How can we prevent those recurrences? Is there a way to prevent it? Is there any medications? Is there a procedure? Or other than just simply watching them? Unfortunately, to date, we don't have any FDA-approved therapy in adjuvant setting, and part of that is a challenge here, and those are sort of the challenge highlighted in yellow, that this is not a common disease, and most of the time when adjuvant therapies get approved, there are larger trials, randomized trials, comparing to active surveillance alone, and see if you've made any difference in recurrence risk. Or is the drug that you're using to reduce the risk of recurrence, is it safe? Is it, what's its toxicity index? So those are some of the challenges in uveal melanoma, but Dr. Carville and Dr. Patel and everybody in the community of uveal melanoma are making strides using gene expression profile guided adjuvant therapies that allow you to seek the signal, expand the study so we can make some differences and reduce those recurrences in these patients. So these are some of the summaries of the previous trials. Again, the benefit, wasn't to a point where it would lead to FDA approval. So I'll just move along. And then this slide is a bit out of place, but I'll give you a little bit of background. So BAP1 is a gene that's commonly mutated in uveal melanoma. And when this BAP1 interacting with HDAC, so one of the terms that was mentioned a little bit earlier by Dr. Sullivan, it in concert with HDAC4, it allows progression and a bunch of other functions that lead to increased risk of recurrences and so on and so forth. Using this drug, cuzinostat, which inhibits that HDAC4, and by doing so also interferes with the function of BAP1 mutated protein, it can actually reduce sort of the malignancy burden or shrink the growth of tumors. And this is a mice model shown here at the bottom. If you forget the graphs, you forget these colorful slides, just look at the bottom 3 panels that are looking at 2 uveal melanoma cell lines implanted into mice, and then they were given either the vehicle or HDAC inhibitor, and you could see that there was a decrease in the size of the tumor itself. So there is a function of this drug, at least in an animal model, that maybe we can leverage into human population as well. And this is a small trial, phase 1 study of cuzinostat in various solid malignancies, again showing that this cancer, matter of fact, does have activity, and we may be able to leverage it. So based on these 2 sort of key points, there is a planned adjuvant study. Hopefully that will be opening up fairly soon, and that was my reason for highlighting that particular BAP1 story. So now, unfortunately, since we don't have any adjuvant therapy, so the high-risk uveal melanoma will have recurrences issues. What are their prognosis? And unfortunately, it's fairly dismal, but there is some hope on the horizon. Okay, so now we're looking at sort of 2 separate set of patients, individuals who have liver-only disease, or majority of the disease in liver and maybe outside the liver is very minimal. They are candidate for liver-directed therapy. It's a it's been a mainstay of uveal melanoma therapy for a long while now. And there are different types of treatments that could be directed at the liver to improve the tumor burden in the liver itself. And it's a long list and ever-changing. But one of the therapies that I'm going to highlight is the hepatic perfusion, percutaneous, and this is a recent, I think 2021 ASCO. This data was presented for percutaneous hepatic perfusion with melphalan for patients with ocular melanoma and liver metastases. And this is the phase 3 trial results. And recently, Dr. Patel reviewed the results of it too, and I listened to it. It was amazing. Thank you. So this was actually, the trial's been going on for a while, and there were initially, for the first study that was planned as a randomized, there was a few issues with it, and it needed to be modified, and FDA guided that modification. I think there was deaths on the earlier part of the study, and then a new filter was designed to allow a more safe infusion of the drug, and then also the 301A led to just a single arm study rather than a randomized to best accessible care. So this study looked at 144 patients, and 102 of them received the PHP, and 42 received the best accessible care. And when you looked at the response, there was about 33% response in the PHP arm, and best accessible care had only about 14% response. There was also difference in almost threefold increase in median progression-free survival benefit. Most of the toxicities that were related to this PHP infusion were more hematological, and they improved without any long-lasting effect. So this looks to be a quite promising therapy for some of the patients who may not be candidate for the subsequent therapy that I'm going to mention to you. So this is looking at a complex picture that I think it's come up, or some iteration of it, at various points. This is the only slide I have for targeted therapy. So all those spots where you see some narration with sorafenib or selumetinib or trametinib or other drugs, those the tried points of interruption in the cell machinery to decrease its ability to divide, grow, and move around other places. Unfortunately, so far we haven't seen anything promising like BRAF mutation guided inhibition in cutaneous melanoma, or but again, there is hope. There is hope. So there is ongoing clinical trial from it's IDA 196 in patient with solid malignancy harboring GNAQ and 11 mutation, and the company released some early data that shows very promising results with 100% disease control with a combination of two different drugs. One is darovasertib, and the other one is crizotinib, which is FDA approved in lung cancer already. So we're using sort of or trying to repurpose some of these drugs and bringing them to a newer disease, and looks very promising. The other trial I wanted to highlight is Foghorn FHD286C001, which is we're a site up for this trial along with Dr. Patel's MD Anderson. It's a phase 1 study for uveal melanoma alone, and the basis of this particular drug is that for cells to grow and move away from their sort of original home, they require gene access, and that gene access is done through chromatin remodeling because these genes are packed into chromosomes and whatnot. So when you access these certain genes, they can drive the disease process. In that machinery, there's BRM and BRG genes that are quite important, and FHD286 inhibits that machinery and doesn't allow access to certain genes. And that graph on your far right with label day 3, you can see that uveal melanoma and AML cell lines were the most sensitive to this, and this drug is currently being tested at in a dose escalation way in that particular those two particular diseases. So our hope is that this will be one of the drugs that we can use to treat our patients, hopefully soon if, in the future. So far you've heard a number of things about immunotherapy, and we obviously going to talk about immunotherapy in uveal melanoma context as well. But unfortunately, what I would like your attention pointed to is here. that uveal melanoma tends not to be very immunogenic or inflamed. Even if it's inflamed, the degree is quite low compared to its counterpart in skin melanomas. So again, that presents with challenges when you test these immunotherapeutic drugs that are quite effective in cutaneous melanomas or non-small cell lung cancers or other malignancies. They're not quite as robust in their response in uveal melanoma. And so I'm highlighting two different trials here. One where Dr. Patel is the senior author. It was published, I believe, last year. And this was using nivolumab and ipilimumab, or Opdivo and Yervoy, in metastatic uveal melanoma. It's a single-arm phase 2 study. The patient characteristics are highlighted on the far end on your left. In the middle table shows that about 18% of the patients can be captured with this particular therapeutic option, and they'll have a response. And out of those, one was a complete response, 5 partials, and 11 were stable disease. And also, again, the waterfall plot and the swimmer plot that Dr. Hamid has taught you about, they're in the panel as well. So not for all patients, but the limitation here is we also don't know which patients we can use these in. So we can avoid some of the toxicities and just pick the ones that would respond to it, and continue and move on the patients that aren't candidate for this option. This was the second similar study, but this was in the first line. So I believe Dr. Patel said he was, they had received the previous therapies, but the Spanish study was in first line setting. Again, results are very similar. So, it tells you that you could potentially use this option for some of the patients, either in the first or second line. And we didn't learn about anything new for Ipi-Nivo, if they had any additional toxicity or not. And we're pretty good at managing that, since we do that for a number of other disease forms. This is probably the most exciting molecule in uveal melanoma to date, tebentafusp, which is an MTAC, bispecific T-cell engager. Essentially, it's a two-pronged molecule. One end binds to the T-cell, the other end binds to the melanoma cell itself. But for this to work, there are particular type of white blood cells you need, and that's called the HLA restriction, HLA-0201. And so this was published in New England Journal of Medicine. I think Dr. Hamid somewhere over there on that list for the work he's done for this. Again, this is the first drug that has shown overall survival advantage in uveal melanoma. The best thing I think I took from this publication was that anybody who received tebentafusp or Kimtac, they had improvement in their overall survival benefit. So, which tells me that we're not really clear how to measure the response of this drug. Does it have to be scans? Does it have to be some sort of blood marker that could help us? Still looking for results on that. Overall, the toxicity for this drug tends to happen early on. And after that, it seems like, and the biggest issue that you have to monitor is a cytokine release syndrome. Is when you get these T cells all engaged, they secrete these chemicals that can potentially cause a number of issues, blood pressure, fevers, heart rate issues, and whatnot. And they need to be monitored in the hospital for the first 3 doses. But they tend, if you can tolerate the first few doses, I think it's fairly easy to move on beyond that. Let's see what I'm at. Okay, I think I'm getting to wrap it up. Sign over there from Dr. Hamid. There is some role for the TILs in uveal melanoma, but it's fairly limited. One of the trials that I wanted to highlight here is the tumor treatment field. It's sort of alternating current devices that are used in GBM, glioblastoma multiforme. It's approved as a second or third line therapy and seen because it shows overall survival advantage. And now it's coming towards melanoma and pancreatic cancer. And this trial will open as a signal-seeking trial first, looking at 10 patients. And if we do see benefit, then they will expand it further. And we're pretty excited about that. And we should open that in the next few months. I think with that, I'll stop. Thank you.
So we're going to transition into our question and answer session. You okay? A couple minutes? Can I ask the speakers to just bring a chair and put it right here? We're going to cover... Oh, sorry, 1 second. Samantha Guild. Sorry. Sam Guild is the key to us having these meetings. And the improvements that we've had in care come not only through treatment, but prevention. And I think that's the most important thing standing here where there's like 2 tanning salons within 5 blocks of us. It's ridiculous that happens. And Sam and her family have not only led this charge of education for patients throughout the world, because these meetings are not only national but international, but have also led the charge of advocacy. And that's something that is the most important. And I think all of us would be happy to stop seeing patients and work on our retirement or our second careers. And that only comes through advocacy and prevention. So we're going to have Sam talk a little bit about AIM, and then we'll do our question and answer session.
So thank you so much for having me. I have to admit I'm actually very giddy because this is our first in-person patient education event in 2 years. We're back. A little limited, but we are back, and I am also incredibly giddy to be doing it with Dr. Hamid, all the presenters here, and with the Angeles Clinic. We've been running these events now for many years, and I am incredibly excited to be here. So I have slides, but I'm just going to be using this one. So the foundation was started in memory of my sister Charlie, who was 26 when she died of melanoma. And at the time, there was only one treatment option for her stage, and the prognosis was incredibly poor. Even I have difficulty keeping up nowadays. It is incredibly exciting. But in the interim, in putting together this foundation, our goal was research. But in the meantime, there's lots of other work that needs to get done, and because of the patients and the doctors and everybody, we have a number of different ways that we are here to support you with the Angeles Clinic and everyone else. We are a team with you. So advocacy, I'll just mention, we were actually the ones leading the effort to ban minors under 18 here in California, and we're continuing doing that. But for education, what do we have to offer? So we heard a lot about the physical needs and how the treatment options are working, and with the doctors to address those. We are also working on the emotional needs that you and your families are facing. And so we have a series called Beyond the Clinic, Living Well with Melanoma, where each month in a podcast we are addressing the various issues that you guys are dealing with. Whether it's dealing with social media and websites and the misinformation, how do you balance the two? Caregivers, how do you care for the caregivers? A lot of different things, and you can go to our website. You can check those out. We also have a physician assistant who works in a melanoma clinic, and if you have questions, reading pathology reports, how do you recognize and manage the side effects of treatment? How do you types of questions to ask your doctor the next time you go in. Again, that's something we can offer. We do symposiums. We have sometimes monthly webinars where we're addressing, as new treatments come out, the landscape is constantly changing, and we have webinars to address those. Those are just some of the things we do. We have a peer-to-peer program to help support you, to match newly diagnosed and their families to veterans. If there's anything that we can do to support you, please feel free to reach out to your doctors, to us. We want to do this together, and together we're going to end melanoma. Thank you.
you can see how Samantha and AIM have really changed what we're doing for our patients and how we're getting through to our patients. It's just very humbling to see the amount of work that's come from not just Samantha, but Val Guild, who was the founder. And I'll tell you, we see a lot of that in our time together, and we're just so happy that we're making these changes for the patients. So at this point, I'm going to say two things. Can I have the speakers bring up a chair and sit there? We'll give one of the mics to the speakers. The other will pass to people with questions. We have questions from Facebook. Can I get the cap on that? I will also say that this will be the last part of our program. As you walk out, there are mugs, Angela's Clinic mugs, because you take something home with you. And we'll look forward to seeing you at our future events. Obviously, AIM at Melanoma is where I ask all of my patients to begin their journey in melanoma. So let's get some questions from those who have remained quiet on the interweb. The first one comes for Dr. Patel. Can you speak to why combo neoadjuvant therapy should only be given 2 to 3 months prior to surgery?
Sure. The combination immunotherapy right now, neoadjuvant, I think as you were trying to make the point, Dr. Hamid, is really a clinical trial option. It's not really an option we're recommending outside of a clinical trial for a couple of reasons. The side effects, when you take combination immunotherapy, 80 to 90% of people will get some side effect. A third, maybe 40% are significant that could land them in the hospital. If you land in the hospital, the chances are high that you might receive steroids. If you receive steroids with combination immunotherapy, you probably have a delay in your surgery date. So combination immunotherapy is stronger at shrinking tumors, but if you give too much of it, if you don't time the surgery just right, you can run into issues with delay in surgery because of these cumulative side effects. So that's the International Neoadjuvant Melanoma Consortium tried to say there's probably a finite amount, and then you can pick up the rest after surgery.
Okay. Dr. Carvajal, we have a patient here who has a great question about, just 2 cycles of Ipi-Nivo and then making a decision. Is that something that you utilize? It's something they've picked up on the interweb.
Yeah, I guess, I suspect that a lot of us practice differently here. I'm sorry about the feedback again. I wonder if it's my phone. But, there are 2 things with immunotherapy that I think are important. One, we've been sensitized to the idea of something called radiographic pseudoprogression, right? And that's because of our experience with ipilimumab, where you treat, sometimes things look worse on the scan when in fact the tumor is just infiltrated with immune cells, and that's why it's bigger as opposed to there being true progression. That phenomenon we saw fairly frequently with ipilimumab, not so much with anti-PD-1, and really not so much with Ipi-Nivo. So because of the toxicity that Dr. Patel was talking about, with Ipi-Nivo, it has been my practice to do 2 cycles of Ipi-Nivo and then scan. If there's frank progression there, then I've changed course at that time point. And that's in an effort to kind of minimize the risk for toxicity.
Okay. I will say that there's a microphone which hopefully won't give feedback. If anyone here has a question, raise your hands, and we'll bring the microphone to you. So right here, this microphone coming to you.
Yeah, I have a very basic question. Is it different when you're talking about immunotherapy and there's something that starts with BRAC? Are you talking about medication? Are you talking about Bractovy and Mektovy on the one hand and immunotherapy on the other hand when you say using medication versus immunotherapy? Because you keep using the word, and is that what you mean?
Yeah, so when I refer to I use the term BRAF targeted therapy, that I use it as a very general term, But specifically, what I'm referring to are the 3 BRAF MEK inhibitor combinations that are FDA approved. So those are dabrafenib trametinib, which is TAF-LAR mechanism, braftovi and mektobi, otherwise encorafenib and binimetinib, and then vemurafenib and cobimetinib, which is zelboraf and Kostelic, which we don't use as much. And so I can't remember the brand names very well. But those are the 3 BRAF targeted therapy regimens that are approved. And so I usually say BRAF targeted therapy just to include referring to those types of treatments.
What is the difference between them?
The difference between that and immunotherapy?
No, the difference between the drugs.
Oh, yeah. So.
I've been taking this bractovi and mektobi for years, and I think it stopped working. But,
Yes.
But now there's 2 others you mentioned.
Yeah. So one of the things we were hoping when braftovi and mektobi would get approved was that maybe it would work a little bit better than the other ones. I think the only thing that's different between the 3 doublet regimens is the side effect profile. And braftovi and mektobi probably has it's sort of the easiest to tolerate from a patient standpoint of those 3. It doesn't have to be. There are slight variations in terms of the side effects, but in terms of how effective they are, they're not dramatically different.
Is fatigue a very common one in the bractovi?
Yeah, fatigue's probably one of the most common side effects of any cancer medicine for 2 reasons. One, these types of pills can certainly cause fatigue, and 2, cancer causes fatigue. And so if you're doing clinical trials and you're categorizing the side effects, when we sort of try and attribute, is this from the cancer, is this from the treatment, but very commonly it's hard to distinguish. But fatigue is common with all of the 3 doublet BRAF MEK combinations.
Okay. So I'm going to ask you a question about...
Thank you.
You're welcome.
That comes from those patients on our live. How long do you usually give these targeted agents if you have a patient with metastatic disease? When do you start to think about stopping?
Yeah, it's a really good question. There's data that about 15 to 20% of patients will have long-term disease control if this is the first drug they receive. By long-term, the longest data we have is like 5 years. So there are some patients who I've treated for half a decade or more with these types of treatments. There is some data to suggest you can consider stopping if patients are in a complete response, and that some of those patients will not recur. We've had a few scenarios in our system where we've kind of utilized that data, talked to patients, talked about the risks and benefits of stopped, and then the disease came back, and we felt very bad about that. And so our policy now is generally to treat indefinitely if patients are tolerating it and it's controlling their disease. That's not true in the adjuvant setting. In the adjuvant setting, which is after surgery to improve cure rate, we treat for up to a year.
Okay, thank you. This is another one that anyone can choose to answer. Do you use PD-1 staining, lag staining, anything like that to choose what you would begin your therapy with? And we can have multiple people answer if you want.
I think in practice here, we don't choose to, because the data hasn't really borne out that any of these stains would help us select those patients who are going to respond to these treatments or not have a response. Most of the, PD-1 stain, CTLA-4 or LAG-3 staining isn't sort of part of the workup prior to decision on therapy. So, if a patient is fit, has the disease, the expectations are, go ahead and start treat them rather than what their tumor markers are showing.
Yeah, I would agree with that. I don't think we have not routinely done staining, although there's some data, sort of a subset of data analysis from that newest study, Relativity 047, would suggest that PD-L1 staining could potentially be useful, but it'd be nice to have a little bit more information before sort of incorporating that into upfront decision making for a patient to say get the nivolumab relatlimab combo or Opdualag versus nivolumab or pembrolizumab.
I'll say At MD Anderson, I am using it. I am using tumor markers. If somebody's newly diagnosed and metastatic and we have a moment to catch our breath and think about options, they have several frontline options. They have single agent, they have combination, different flavors of combinations, and I'll use the PD-L1 stain to help me guide. So a PD-L1 negative tumor, the subset data from multiple studies suggests that they do better with combination therapy. And then it's a sort of an internal debate which combination therapy. But if they're PD-L1 expressors, anything more than or equal to 1% on the surface of their melanoma, PD-1 monotherapy will do pretty well with pretty much equivalent in terms of statistical survival and statistical progression. So, or maybe not progression, but statistical long-term survival. So, to spare people toxicity, as somebody who has 1% of this marker on their surface cell, I would use that. So it's true we've never had a proper study to kind of compare negative, positive, and combination mono head-to-head, but I think the data is intriguing enough to start using it.
Yeah, I think these data, it's important. It colors the discussion, but it doesn't provide kind of prescriptive, this is what we have to do because the tumor stains for this. But it definitely colors kind of the decision-making that, as patients and caregivers you have to make, like what is your risk tolerance? What are our goals? And that's really important, right? If the goal is cure, then maybe our risk tolerance is a little bit higher. But so the conversation is very, very complex. I think it's just the staining adds to that complexity.
Okay, Dr. Boyko.
Follow-up question to this, and make it more general. Do patients do it? All of these great therapies, they won't presuming that T cells can get to the tumor, can infiltrate the tumor. What if the patient's tumor is T cell cold, T cell cold? This patient first goes the screening to decide whether or not he's going to benefit from cell therapies, and what do we do if the patient's tumor is cold? Do we still put him on these toxic immunotherapies?
Yeah, it's a great question, and I think there's a lot of interest in trying to make these tumors that have excluded the immune cells from them to make them able to receive the immunotherapy and have it be effective. There were some data to suggest that in terms of the two leading checkpoints, that CTLA-4 actually could drive immune cells into the tumor, and that would be some rationale for the combination, why the combination works. But I think it's an open question. We can do things directly to the tumor. We can inject stuff into the tumor. We can radiate the tumor. We can freeze the tumor. There are things we can do directly to the tumor that may enhance the inflammation of that tumor, but it's a pretty wide open field at the moment. I think that's why some of these neoadjuvant studies are so interesting, because in that paradigm, you treat and then you have this resection specimen to look at, and you can evaluate a bunch of different strategies in rapid fashion to see if you get this infiltration. The imaging that Dr. Hamid mentioned may be able to do it without surgery to identify when you're driving cells into the tumor sites. So I think that's going to be a great open question for trials that are coming along.
Okay, one for Dr. Ganji. You discussed metastatectomy for patients with metastatic melanoma. Can you expand on that, give us some situations where that would be a viable option for our patients?
Sure, depending on the degree of the disease that a patient may have. There are some patients who undergo a variety of different therapies and may have progression in a solitary lesion or solitary location as a metastasis within their body. So if they're not responsive to therapy in that particular area and we have no other reasonable options, we will consider metastatectomy at that point, given that all the other sites may have responded. If you have limited disease at one site, it's also a consideration. And obviously, historically, there have been studies looking retrospectively at patients who have benefited from metastatectomy in select settings. And now, with the advent of a number of additional therapies, we would consider it in those patients.
Great, thank you. I have one. I think Dr. Sullivan can answer this because there's multiple questions that have come forth. It's about immunotherapies, though. What if you have immune suppression like CLL? What if you've had a transplant? What if you have multiple sclerosis? What do we know about giving these drugs to those patients?
We don't know a ton. With that said, there's, I would say, two things about the immunosuppressed population. First, it's probably likely that if somebody's on full-on immune suppression, they're likely to have a muted response to immunotherapy. It makes sense. It's sort of like giving the antidote to the immunotherapy. What's interesting about that statement, however, is if somebody doesn't start on immune suppression, starts therapy, has a remarkable response, but then needs to get immune therapy down the road because they develop a bad side effect. Sorry, immune suppression because they develop bad side effects. It doesn't seem that everybody has a reversal of their benefit. So there's a lot we don't actually know about. And so ultimately, if somebody comes to me and they're either on immune suppression, one question is, why are you on immune suppression? Is it because they have a liver transplant? That's a really hard situation, because if I give that patient immune checkpoint inhibition and their liver goes away, they die from that. And so I think we treat patients who are on immune suppression if it's and we follow very, very closely with the doctor who's managing whatever condition they're on immune suppression for. We do our best to modify the immunosuppression so that it's the least immune suppression that we can think about, or more targeted to the disease. For example, we try and get people off of steroids or down on steroids and onto other types of treatments because steroids seem to be more generally immune suppressive. But the bottom line is, these are the best therapies we have, and for patients, it probably offers their best chance of getting rid of their cancer. And so we will treat patients in that scenario, but it's very complicated and can be extraordinarily challenging. With that said, in the adjuvant setting, it may be a reason for me not to treat somebody and to say, let's hope that surgery did the trick. We'll follow you very closely. God forbid the cancer comes back. Then at least the decision's a lot easier because you're not going to be cured by surgery now. And so the risk-benefit ratio sort of favors treating at that point. But I don't know if anybody else wants to tackle that. No, yeah.
Okay, we can go. So if anyone wants to tackle when they would use BRAF inhibition as a first-line therapy. And it doesn't have to be you because you spoke on it, but...
Well, we had an interesting discussion last night about triplet therapy, which in this context means BRAF, MEK, and a PD-1 inhibitor. There is a triplet regimen that's FDA approved in the frontline setting, but there hasn't been wide uptake of use of that in the oncological practices. Mainly because I think there's this fear of, after you use that, what do you use next? You've kind of used every drug and thrown it all together except for ipilimumab. So, but interestingly, if you look, it has some of the longest intervals. Patients stay on treatment a long time with a median progression-free survival of 15 to 16 months. Toxicity is a concern, though, more is more. So what I think the takeaway from DreamSeq for me is, if immunotherapy starting first does better in the long run, combination immunotherapy, but there's a small subset of patients who actually die very quickly because immunotherapy doesn't work fast enough for them. They never make it to cross over to BRAF and MEK. I think if we can do what we can to identify who those rapid progressors are, those are patients where you might start with BRAF MEK first, or you might start with some type of triplet therapy.
Okay, thank you. Do we have any other questions? Hold on.
I don't need a microphone.
Okay, I know you don't need a microphone, Alan.
What percentage of the population has BRAF?
So what percentage of the population has a BRAF?
It's interesting. If you look at skin melanoma, it's about 50%. If you look at mucosal melanoma, which are the melanomas that are on the lining surfaces, like in our sinuses or nasal cavities or genitals or our gut, it's about 10 to 15%. If you look at the melanomas that arise on our palms and our soles, it's about 10 or 15%. If you look in the eye, it's like 0%. So it depends on where the melanoma arises from, but if you look at all melanoma, it's about 40 to 50% is usually the number we quote.
My second part is Yervoy the same as Ipi?
Yes. That's the easiest question of all day.
Yeah.
Thank you, sir.
I saw how quickly you chose to answer
you know what?
that
If it could be quick, yes.
This question was sort of a personal to my case. I don't know if I'm allowed to do that.
We don't answer personal questions. If you can make it a broad question.
I'm trying to think of how to make it broad because I probably should be dead. I don't know.
Okay, well, there we go. We're going to stop and maybe we can have a one-to-one on that question.
I'm just saying, I mean in terms of what I've heard, in terms of therapies lasting 13 months, 16 months. You know.
So I think what you're bringing up is a really important point.
I've been on these. Is BRAF
Yeah. So I think the.
I've been on that like 2.5 years, 3 years.
So to generalize your individual point, statistics are statistics. They don't actually, so if we say the median, it means half the people before that didn't meet that, whatever that is, relapse rate or be alive, and half the people did. If we talk about endpoints like the 5-year whatever, relapse-free rate, progression-free rate, survival rate, that's just that. It's how many patients at that time point are still relapse-free, progression-free, alive. So it's, and I think that's important to know that there are outliers. Actually, a lot of times now the outliers are the patients that aren't having the long-term benefit, the patients who are quickly succumbing to their disease. Most patients are living much, much longer, and they're remaining on therapy much, much longer. And that's a testament to how the field has moved forward. The problem is too many people aren't. And so that's the whole point of the last few talks.
Historically, there's a certain point where that brachytopia just stops working. Is that normal? That it
doesn't have to. I mean, the bottom line is sometimes, as people are on it for longer periods of time, I have a patient on for 9 years.
But I think a good point to end on is that that's not the end, that we have so many different options. And a patient who would say, well, I've gotten many years on this, and now I need something else. There are newer drugs that target that pathway. There are adoptive T-cell therapies. There are bispecific antibodies. So there are a multitude of things that are being looked at, not just in cutaneous melanoma, but also mucosal melanomas, ocular melanomas. There's TIL for ocular melanoma, and also patients with brain metastases, etc. So that's a great point to end on, that how clinical trials have expanded our scope. And perhaps next year when you come, you'll hear about more FDA-approved therapies. So with that, I want to thank everyone for coming. There's, I think, still some coffee and pastries. Please feel free to take some with you. There are Angeles Clinic cups, so keep in the front of your mind. And I want to thank all of our speakers who have come today. It's been just great. Thank you.